interesting tips and tricks and observations that we can all take home with us. And so let's see. With that, what? Yeah, you have something to say.
How about that smoke? No, just actually one small housekeeping point is that as we go through the talks this afternoon, please feel free to write down some questions as they come to your mind and either bring those up towards the end of the session when we have our closing roundtable, in which we'll invite all of the speakers up to the platform for some questions. And if you prefer, staff from ESCMID, namely Carla Seiler and Chiara Speziale, will be circulating with some question cards. So if you'd rather submit your question that way, that's totally fine. We'll be happy to try to get to them if time allows.
Okay, so I just had an idea, because there's a lot of people in this room, and there's a very bright light, by the way. But so we do have a few minutes before the session begins, and so would anybody like to share what they would love to hear in this session? Some things that are on your mind. So we're going to have again what I think is going to be a great talk here from Jen on challenges and opportunities related to human talent. We're going to hear two, I think, very interesting perspectives from industry on, their particular paths with current agents through preclinical and into clinical development. And then we're going to hear from our regulators, FDA and EMA. And but anyone have anything that they hope they hear, or since your speakers are all here listening, that you would like them to consider when they get up here, as opposed to sort of reacting to what they're going to say to you? So I will start. You know what? So I think my name's Erin Duffy. I work with Carbex, and Carbex supports people like those in this room to discover and bring through first in human new antibiotics, preventatives, and diagnostics. And if you took a look at our portfolio as it exists today for therapeutics, let me just focus on that for a moment, it is heavily weighted at this point towards non-traditional therapeutics, whether they're antivirulence agents or therapeutic phage or monoclonals or other non-traditionals, non-BLI potentiators, etc. And a big question is always, how do you most expeditiously develop them to showcase what they do? So you have an antibiofilm agent. What's the best way to develop something like that expeditiously? Can you use a limited pathway type approach, and what would that look like, and what are the other factors that you would need to consider? Because there's this push-pull all the time, and we just had a little game theory exercise yesterday, which is, do you plow ahead with an indication that is going to be, how to get there, and probably not a great label, but you can get on the market and maybe do a descriptive phase 2 along the way, or a proof of mechanism study? Or do you be patient and do that other study? And what are the trades in that? And I'd really love to hear from our two regulators about those kinds of things. How about you? You probably have a question.
Well, I think one of the key points that we'll be looking for to be addressed, and it will be addressed to some extent, I believe, from 2 or 3 of our speakers today who are in industry or have spent time in industry advancing various programs to the IND stage as well as to NDA and post-approval, is really about the translatability of early clinical and as well particularly non-clinical results that would drive certain programs to an IND stage and then other programs to be sidelined or turned off.
So here's a question. Let's have a show of hands. How many people think innovation is alive and well in the AMR space? Can you see? So that's not as many hands as I thought, actually. It's pretty good showing. But so for those of you who don't think innovation exists, why not? What's missing? Don't make us call on you. Yes, Tim.
Call it out.
So I was being very specific. You said alive and well.
Okay.
So I don't think that innovation is well. So I think that innovation is highly, highly challenged. And it's certainly not in a position where it's robust in itself. So alive, yes. So we agree with that absolutely. No question. But I think the situation is at a point of critical weakness.
And what, since you're on the hot seat now, Tim, so there are potentially many factors that contribute to that weakness. What are your top three?
Well, obviously, it's about the sustainability of the pipeline. You know, how much financing.
Thank you for playing, by the way.
Indeed. So firstly, I'd say it's about sustainability of the pipeline and the ability for there to be execution throughout the R&D time to discover, develop, and deliver new products to the marketplace. It isn't sustainable yet. Maybe we've had some nice improvements with things such as Carbex and other things, but nonetheless, we're at a point where it's not clear that the situation can be on sustainable footing. So that's number one, sustainability of the pipeline, therefore appropriate financing to support the research and development activities that would happen in that. I think as well, the topic that's going to be discussed, we need to think about the skills and capabilities to do the R&D. And maybe we're going to hear that we're already at the tipping point. But it is certainly a concern as well that we have, you know, all the great people in this room and a few who aren't able to join. But have we reached a point where we don't have critical mass, and certainly not critical mass to prosecute on R&D programs over the decade plus that it takes for that to happen?
Okay, that was two.
Let's
Extra
just
credit, three.
Let's just focus on those two,
Okay,
right?
okay, so, but you don't feel, for instance, I mean, you know, there's sometimes a criticism that maybe we're not developing the right products.
Oh, okay, I get the clue. No, I mean, it is always a challenge in terms of, are we understanding the unmet needs and therefore the types of products that are wanted to be developed to address those unmet needs. I could very lazily say, oh, yes, that's what one third thing we need to do is be clear that we're focusing appropriately on those unmet needs. But knowing that it's actually very hard to specifically define that in such a way that it, you know, it's not a cookbook recipe approach. And it does require not only a lot of crystal ball gazing, but thinking more broadly and having broader approaches. So,
Thank you.
Samir Qadri. I am a critical care physician at the NIH in the U.S. And I look at innovation from a different lens, right? I'm in front of a patient at the bedside. And what I'd like to state is, it's obvious, but I think it's worth stating that from my standpoint, I'm having to learn the name of a new cancer drug, a new immunotherapy drug on a weekly basis, which I think is a good thing for that field because there's so much activity, not only in the development, but in the ability to successfully bring that to me. And that's not true, obviously, in the case of antimicrobials. And again, I'm preaching to the choir here, but I can tell you that the pain of not having good drugs for our patients is felt even today, and perhaps the inequity in the development and bringing the drugs to the market for antimicrobials versus the other drugs that are used is very stark.
That's insightful. Oh, wow, we're getting them now. Okay, Carmen.
Hi. Yeah, I'm just curious, when you talk about the lack of innovation in AMR, are you talking how, like, in comparison to what? Just because, so my partner works for a biotech company, an AI biotech company, and the reality, in my opinion, is that the AI is not really delivering. It's not really adding anything. And they work in a field where they're just churning through the same targets as everybody else. And if you look at things like neurodegeneration, there's a huge investment in that space, and nothing, is really coming through. I know there have been a couple of new drugs that have been put on the market recently. The MHRA approved one a couple of weeks ago, but actually innovation in a lot of different areas is not really there. So I'm just wondering, when you're talking about the challenges in this field, like, is it something that's, I know it has its own unique sets of challenges, but maybe it's more of a global problem, because PhD students get paid something like 30 grand a year, which is 3 times what I was paid when I did my PhD. But then they leave and go into finance. They leave and go into anything other than academic research because it's a really horrible job. It's really hard. It's just brutal. It's challenging. You have to kind of follow what's popular and what's getting funding, not necessarily what's of value. It's so tough. You have to move all the time. And so I don't know, the sort of challenge around innovation and funding in AMR. Do you think it's a unique thing, or are we talking about a much wider problem?
This could be a fantastic question to explore during the talks, and particularly during the roundtable. I mean, I think, of course, you've laid out some very pertinent and immediate challenges to the space. I think if you look at the words innovative or innovation, that also could have some very specific meanings. We've all seen reports and scorecards of different agents, whether those are in, for example, the preclinical pipeline, and their innovative attribute or character is sometimes scored. We'll hear later this week, I think, from a number of speakers talking directly about that with regards to whether an innovative quality in and of itself is a key attribute, an important attribute, or whether an antibacterial agent that is safe and effective and meets the quality standards is actually sufficient regardless of the innovative character.
Yeah, I mean, often when it's said in relationship to the products that have been approved over the last N years, where let's call N maybe 10. I always think that's a little bit unfair, actually, and partially to the point earlier just about the numbers of things that come through the AMR pipe versus other therapeutic areas, but that's more in translation, to my mind, rather than sort of the innovation, so how to bring that forward. But I just raised it because it's often an indictment, and it bugs me. Yes, intended. Yes, thank you. We actually have 2 more, and then amazingly, we're going to be at our time. So, yes, Valeria.
Thank you, Chair. Valeria Gigante da Bruccio. Just to build on the previous comment, just to give you some figures, actually in one region in 2023, there were approved 20 new products for oncology. In AMR, between 2017 and 2023, we approved overall 16 products. So really, the numbers are striking. When we talk about innovation, the pipeline contains some good innovation. There are some innovation. However, the problem is that they're not long-lasting innovation. We really need solutions that are game changer, that are definitive solution. Otherwise, when the product will reach patient and marketing authorization and broader clinical use, we will already have seen like resistance emergence. And when we will try to study the same product in children, we will, after 10 years, probably the resistance will be skyrocketing. So this is why we need innovation that are long-lasting. Thank you.
And often the case, sir, you get the last word before we start.
So, John Rex, for those of you who don't know me, delighted to see this come together. And I want to point at the thing that makes anti-infectives weird, which is that they're kind of like, we'd like them to be like orphan drugs. We'd like to we'd have rare settings, hopefully, but we don't give them the characteristics of orphan drugs in other spaces. So in an orphan drug for a genetic disease, there's the pricing models are different, the numbers of patients are tiny, but it's a totally different mindset. And so when I think about the conversations here, when I look at a company saying, should I pursue this? To me, that's the question. Is this actually something that is worth spending the next 10 years of my life on? One of your questions needs to be, how do you access that special characteristic that makes it, right now they won't technically be, most of them won't technically be orphan drugs. A new drug for Shigella will not be an orphan drug, I don't think. But there may be characteristics of it that you can bring out. And also, I think there's a larger community conversation about what actually is an orphan drug. Yep. So that's something that I would like to hear discussed this afternoon. I think you've got an interesting start with why people are leaving, but for me, the reason to get people, the thing that would get people back in would be solving this problem on the back end. I spend a lot of time on pull, on getting governments to reimburse, but there's part of it that's our challenge too, which is to articulate really interesting and exciting products and why they're interesting and exciting. So thank you.
And a really great point. And actually, we are now live. So Jen, we'd love you to begin.
Is this on? Okay. Hi, good morning. So I just want to preface what I'm going to speak with you about, listening to some of the conversation that's just gone on, by saying that I have spent the last 6 years not in the anti-infective space. So when Novartis shut down ID in 2018, I moved into global business development, and I had headed, up until 2 months ago when I retired, I headed business development for Novartis across all therapeutic areas on the West Coast of the US and across Canada. And I saw every other therapeutic area except ID in that role, although I did do all of our out-licensing of our infectious disease program, so I did see that. So I've spent the last 6 years looking at every other space, and I spun out companies, I've done investments and in licensing and out licensing of assets at all stages of development. I spent a lot of time in radiotherapy, so radioligand therapy, a lot of oncology, and a lot of neuroscience. And so listening to all of this, it's actually kind of interesting because I have a lot of thoughts about what happens in those spaces from an investment perspective, from a clinical development perspective, and from a business development perspective that doesn't look anything like ID. So it's a little bit heartbreaking to have seen that for those last 6 years and then do the compare and contrast. And there's a lot of things I can say about that, but I'm not exactly talking about that. So I'm going to set that aside for the moment, and I'm happy to talk to anybody about that experience. But right now, I was asked to speak on, and can I do this? Is this? Yeah, okay. I was asked to speak on the challenge of the talent drain. So again, going back to 2018, so we had started the infectious disease area in Novartis in 2003. We had both antibacterials and antivirals and some antifungal for a little while. And in 2011, we moved the whole group out to California from Cambridge. And I was the only member of the ID leadership team that made that move. So I got to rebuild the whole group out in California. It was really great. We had great antivirals in the clinic. We had great antibacterials. And in 2018, Novartis decided to get out of the anti-infective space. And so I had to shut everything down, wind everything down, spend time out licensing the programs. Fortunately, was able to place all of our talent in other roles, and I'm going to talk a little bit about what happened with that talent. And so that's kind of the subject matter of my talk. I know I don't have a lot of time, and I'm going to skip over a lot and try to focus on things that I think are going to be most relevant given what we just had this discussion about. But I just wanted to quickly say, at the time that we were shutting down, we actually had a really great pipeline. And the antiviral pipeline was about the same number of programs. We had roughly the same number of people on both sides of the fence, so things were going well. We had compounds in the clinic. We had programs all the way at the start. We did focus on small molecule in the antibacterial space, but things were going really well. And this was just the biology group. At the time that we were shutting down, there's a couple people that are missing there, but this was basically those that stayed in the antibacterial space after we shut down from within that dedicated antibacterial group. And actually, one person has since left the antibacterial space, so we're down to 3. But the situation is actually worse than that, because we also had many, I don't know if there's a pointer, maybe there is, many people, something in the neighborhood of about 100 dedicated to antibacterials across all the areas of science and medicine that go into that. And again, it's only those 3 that I showed you on the other slide that actually stayed in the infectious disease area in the antibacterial space. So we lost 100 people plus, and that was just from the antibacterial side. So that was really painful. The burning question that was on a lot of people's minds was, am I going to stay? I mean, at that point, I'd been a bacteriologist for almost 30 years and had been in the infectious disease discovery and development space for close to 20 at that point. I love it. I absolutely love antibacterials and infectious diseases. We are actually quite fortunate to have really great model systems. I mean, after spending time in all these other areas of science and medicine, I can firmly say that antibacterials has the best model systems. We have a really great likelihood and probability of success if the phase 1 looks good. I mean, you can pretty much, if you've got safe and predictive PK, you're probably going to have a good drug. The field is very supportive, right? Everybody's here, everybody's been trying to support each other. And because we tend to be smaller groups within big pharma, we get to do roles that lots of people in other areas of science and medicine don't get to do in big pharma. Like, I did everything because we didn't have, a dedicated person for every single aspect of the entire end-to-end research, development, commercial. So did a lot, saw a lot. And obviously our efforts are globally important. But on the downside, when I was deciding, am I going to stay in ID, am I going to leave, I have to say, it's exhausting. It's very mentally and emotionally exhausting to have to tell a group of people, you've done phenomenally well, we have these great successes, we have compounds in the clinic that are addressing high unmet medical needs. Thank you very much. No thanks. So very, very taxing on the mind and on the body. And I was asked a number of times by organizations that were potentially looking to hire me into a role that I was in or a similar role. Well, what would you do differently if you came here? How would you make this successful? And I had to laugh. I was like, well, if I had that answer, I would have done it at Novartis, right? I mean, it's not like I kept all my best secrets, until I spent 20 years and then went somewhere else. I mean, it's hard. It's a really hard space in many, many ways. And the talent pool is small. So, we kind of struggle to get really great people to come in and stay in. So it's a small pool to start with. And then, trying to convince people not to leave the space, which is hard to do. I certainly think, the investors are either not interested or misaligned in many ways. And then, we literally have to deal with the politics. So that makes it really difficult. I decided to leave, and as I said, I went into business development, but I stayed connected. At the time, shortly after, there was this article then written about the talent drain. And this group came together and pulled a number of us and some smaller organizations as well and said, so tell us what's happening with the people that were in your organization and where are they going? So this kind of started a whole thought process around the talent drain specifically. This was the publication that came out, and really there's just like a couple of key messages and charts, but basically you have a major drop in the number of publications and a major drop in the number of scientists contributing to the space and to the publications. So this was something that was really notable in comparison to a field like oncology. And it really struck people, struck a chord with, wow, we're really losing information, we're losing talent, we're losing institutional memory and organizational memory. And I mean, just from a numerical perspective, you've got publications going down, by basically half, and you've got 35 times as many publications in oncology than you do in ID, and more than an order of magnitude more researchers in areas outside of antibacterials. And the problem is that now you have a smaller number of people available to work in this space, whereas we need to keep moving these programs along. So that's what the report really came to understand. And they poked around on LinkedIn. They did some more digging even beyond the publications. And what they learned was that this was actually from a number of small firms. So this was, I think, 6 small firms that had to shut down. And where did their talent go? So out of an original number of 152 people, they're down to 12 still in the antibacterial space. And I already know that there's some of those folks have since left. So this is even a couple of years old now. And there's just a drain. There's just physically a drain out of the space. So the challenge here is that there's a lot of spillover effect. And there was a physician who just spoke with us in the room. I mean, when you also have fewer residents in infectious diseases, a lot fewer. I think something like 40% of last year's residency class was filled out of what should be 100% of attendees. So when you don't have those docs, you don't have trial investigators, you don't have consultants, you don't have translational medicine experts coming in to help develop these drugs. And without the R&D experts, you then continue to have fewer and fewer mentors. You don't have as many publications. You don't have the journal reviewers. You don't have study section members. I mean, I still sit on study sections, and I wonder, can't you find younger people to do study sections? But it's really tough. So you also have these gaps where you can't fill in for the people that are trying to do non-traditional work, like Erin just mentioned. So it's hard enough to find people who are still talented in more traditional antibacterial approaches, but now that you don't even have mentors, to try to help the non-traditional space move along in an even more, challenging discovery and development environment. Investors, right? There's very little incentive for an investor to stay in that space, and because there's a lower chance of a big upside, you have difficulty keeping people, in small biotech and in the investment space in antibacterials. So, this just continues to spill and spill in such a way that you now have fewer people who are able to speak outwardly. There are few people who are experts in some of the policy-making arenas. You have fewer people who are experts who can speak to the public about what infectious disease, challenges are, both on the patient side and on the drug discovery and development side. So I was asked to propose solutions, despite me just telling you that if I had all the solutions, I would still be doing this job at Novartis. But really what it comes down to is a couple of things, and that is you have to be able to build the talent pool and the incentives from 2 directions. You have the physicians who are in clinical practice using these agents, So we have to fill those residency classes. We have to incentivize ID docs to stay in the space, to get into the space and stay. However that has to happen, you know, forgive their loans, have some kind of way of keeping the ID physicians in that space. We really need a knowledge hub. We need a place to aggregate. We need a place to come together and be a community, whether it's virtual, whether it's a meeting, a bigger meeting, but there needs to be a room with everybody in it. You know, from an investor and a company perspective, I think we have to think about new ways of compensating people who go into the business in order to stay in that business, because again, from my BD work outside of ID, I can tell you it's really hard to say to somebody, well, you know what, you should stay in ID versus going to this oncology company where, you know, you could wind up having your shares be worth quite a chunk of change. So maybe we need to incentivize different ways that are not just equity-based, because the promise of that equity turning into something really substantial is very, very small in this area. We really need to encourage publication. We really need to encourage presentation. We need to have people seeing and reading and living each other's science, because without that, we will do the same mistakes over and over again. We don't know who to contact. We're not sure how models are changing. We don't know whether there's, you know, new ideas, interesting things going on. So, you know, if you're a CEO of a company or you're thinking of forming a company, please make sure that your talent is actually able to externally validate themselves and their science, because it's really important for people to be able to do that. And then, you know, hosting interns, bring people into your company, into your organization, let them learn from the experts, you know, how to be in the space, and keep the burden, the administrative burden of doing that low. Make it real easy to bring in interns who can be learning about the antibacterial space. And then last, but actually first, mentoring. Just, if you're in it, help another person, and just keep doing that, because the numbers are so small that we all really have to be engaged and continuing to be mentors. I mean, like I say, I stepped away from the space 6 years ago, but I have stayed, I mean, I would call it my other day job, and I did it for free. So it's really, really important to be mentoring other people, and encourage and reward mentoring. So if you're in an organization and you've got people reporting to you, or you're, you know, thinking of starting a company, make sure that you give a really strong incentive to people to be mentors. It should be like the first thing on their job description, and you're going to be expected to mentor other people. That's how we will build this community and maintain this community. And this is a last piece that I just wanted to mention, and that is, you know, part of the challenge with antibacterials, and I would say infectious diseases generally, is that it's kind of viewed as dirty, right? Oh, you know, well, we wouldn't have this problem if hospitals were clean. We wouldn't have this problem if there weren't IV drug abusers. We wouldn't have this problem if whatever, right? You name the blame game, and it's out there. So ID is kind of looked as like a dirty kind of space. I grew up in the 70s. Cancer was a dirty word at the time. Like, you would never say it. You didn't want to be next to people who had cancer. That's gone. ID is still that way. So we really need to help the public understand that people get infectious diseases and get, you know, untreatable bacterial infections having done nothing wrong. This is, and the more that we add immunology drugs, and the more that we add new oncology drugs, and we have people undergoing different types of transplants, and they're living longer and longer, infection's going to go up, not down. So, and it's no fault of the individual. So we really need to make sure that there's an awareness that clinical resistance shows up because of appropriate use as well as inappropriate use, and it's not going to go away. So public perception is something that everybody should be involved in educating. And last but not least, you know, I would say stop dabbling and start investing. Like, make the investments really meaningful. Big pharma needs to understand, and they do understand, but they need to act on the fact that, like I just said, if they don't have access to good antibacterials, it impacts all of their business, because the patients who are getting some intrathecal injection, or they're a cardiology patient and they have you know, invasive devices, they have indwelling catheters, whatever, those patients will have infections. That's just a fact of basic biology. And so big pharma needs to be investing because it's impacting their ability to bring their more lucrative products to the market and keep them there and keep them in good utility. And then, I think on the investor side within the ID area, I know that there's a big push on novel targets and novel drugs, but also I think having communications with physicians about what are they actually looking for, what do they need, what's clinically important, and not get too stratospheric on new, everything has to be new, because it's hard. I mean, it should be done, but it's not the only solution. So we need a good balance of some new and some, I know exactly how to use this class of drugs. And also, when you go into new, you lose the economies of scale on manufacturing, which has been a real challenge in this space. So that's, I think, really everything I wanted to say. I just was throwing this out there that I live in San Francisco in the Bay Area, and I feel like talking about AMR is a little bit like talking about homelessness in San Francisco. It's just a very complicated, very lots of tentacles to try to figure out how to solve this. You can patch little holes and you can make yourself feel good that you did one thing, but it's really not a solution. Like, we need a very, very big thinking about how to get through this challenge. And unfortunately, some of those little patches of holes distract from the ability to keep the message going of how important it is to build this space back up again. So fix the market, fix the investments, fix the talent drain, because the science is hard enough. Thank you. Do I sit? Do I stand?
Terrific, Jen. No, that was fantastic. Thank you very much. So we do have a little bit of time for those that would like to pose some questions to Dr. Leeds. And again, please feel free to either submit the questions on cards that will be circulated, or come up to the microphone and introduce yourself, please. But Jen, you've certainly given us a lot to think about. I think your summation of that, the loss of talent report, was particularly striking. Concerning. I think there are some avenues that you've put forth as well that could lead to a solution space. Amongst if I could just perhaps start with one question before opening it up to others. You had talked about the possibilities for internships, and of course those, in our company we've used that approach for a number of years. There were certainly benefits and arguably some disadvantages as well, but it really comes down to training and time and resources for those folks. I think it certainly did represent a fantastic opportunity to let new entrants into the field get their feet wet, and at the same time let the host company get an understanding of how this person works and whether they might be a good fit for the organization. Have you, over the last years, seen some approaches that work better than others, or collaborations that are more likely to turn into someone being retained within the company?
Is this on? Yeah.
It is.
So we had a number of internship programs that were sort of separate from just like the larger Novartis internship program. We had very good relationships with certain universities in the UK, actually, and it was part of a personal professional relationship with one of the scientists in our group. And every year he would bring in, 4, 5, 6 interns specifically in the in vivo pharmacology space. And that worked really, really well, because that's an area where, I'm looking here, that's an area where there's even fewer people training. I mean, it's like, you know, if you think you're getting drain out of whatever, high throughput screening or whatever, I mean, in vivo pharmacology is a really tough space to recruit people into and have them stay in. So we did that, and then we really trained these people up well. We stayed on their thesis committees. We stayed very engaged with them throughout their academic experience as well as the company experience. And then we were able to recruit people, and it was a really great relationship. So in part, I think the quick way to answer the question is, if you have a focused area that you really feel strongly about, and you can stay in the relationship before and after the internship in a really meaningful way, like being on a thesis committee, it really does get you to know that organization, the trainers in that academic institution, and then, you know, sort of handpick the best talent to bring in and retain.
Thank you. Go ahead.
Jürgen Bolita, University of Florida. Thanks so much, Dr. Leeds, for offering this perspective. I have 2 positive comments and 1 question which is a little bit heartbreaking. So one thing which worked really well in academia is to import talent from other areas, including oncology. That worked great. One aspect, we might have a little bit of an overproduction, meaning not an overtraining, not in a very positive way. So there might be more trainees coming out of Australia because we just don't have the industry. So if we had access to that pool, that might actually help address it. And the one somewhat heartbreaking questions I have for you: what do I tell to my PhD student or postdoc who has a small toddler and a family, single earner situation, who wants to go into the pharmaceutical industry? How do you think we can entice these guys to really make the step and take perhaps the risk of going to infectious diseases?
Well, they could move to Indiana, and I mean, Eli Lilly seems to be doing something. You know, I think if you are willing to go into big pharma, which is, you know, different from a smaller biotech environment, the good news is that if you go in and it doesn't work out, like in 200 people in my case, you know, there's movement within the company. So I actually think it's a reasonable risk to take, because if you are talented and you demonstrate, you know, that you're a good scientist, we were able to place everybody who wanted to move and stay within the company into different therapeutic areas, and that was really not a problem. So there's good movement. I think if you're going into a smaller company, maybe one suggestion is, well, you know, try to do it in a region of the world or country or wherever you want to be, that there is some, you know, there's a larger community of biotech or you know, government roles or non-government organizations kind of within the same vicinity, so that, you know, if for whatever reason you can't continue on, at least you've chosen a location where there's some flexibility and some movement.
Jen David Patterson from Singapore. I loved your presentation, but I might almost expand on your response. And do you think it's just part of a cycle that instead of the Bay Area or Boston being the hub of antibiotic development, maybe Basel, Bangalore, Beijing, you know, is it that non-US based antibiotic development is where it's going to be in the next decade, and we just have to accept that for a variety of reasons it's not really working in the US? Or is that oversimplifying things?
So, no, I don't think it's oversimplifying. I think trying to get this to work in the US got harder for a number of reasons over the last 2 years, I would say. So, you know, if it was hard to begin with, it got harder. And again, I'm happy to talk about why I think that's true. I do think that there is definitely a shift in not just the interest level by countries, but the interest level by talent ex-US. So, you know, we're seeing pockets come up, and then we're seeing, like, you know, much bigger efforts come up in other areas. My answer to that is I don't care where it comes from. I just, you know, if there's a country or region of the world that is willing to invest, that is willing to support, that is willing to train, that is certainly willing to discover and develop new drugs, go for it. I mean, it's heartbreaking to as a citizen of the U.S. to see this happening, you know, especially having lived through a pandemic. And okay, like that wasn't enough. Do you realize that there's risks here to national security? There's risks to, you know, all kinds of supply chains. But I do think that having it get rebuilt to a meaningful degree in the U.S. anytime soon is going to be very difficult. So go for it, ex-US.
Yeah, hi. This is Anmol Agarwal from Venus Medicine Research Center, India. So we are a member of AMR Industry Alliance, and I've personally worked extensively on the report that you have just referred.
Thank you.
So in that report, it's you just showed that the publications is very less for AMR as compared to oncology.
Yeah.
So definitely it shows that the research is less comparatively. But don't you think that the main reason is the broken market of antimicrobial resistance? If that is fixed, then automatically the pharmaceutical company would have been able to be in a position to give more opportunities to invest in AMR. And
Yes.
Yes, and if they would be and if they would be able to invest in AMR, then definitely research would be happen more, and the scientists and researchers won't be leaving the industry. Right now what's happening, what I personally feel is that there is not lot more opportunities, research in AMR, and not that the innovation is difficult.
No.
Like you said, the translation is difficult, it's not the innovation, right? So you had a lot many pipeline candidates in 2018 when Novartis shut down. But given that it's the main reason is the broken market, the resistance that comes once the drug is commercialized after 10 years or 15 years, it's no longer working and the market goes down.
Yeah, so no doubt that a major reason for this talent drain is the broken market, 100%. The other thing that gets layered on top of that is that unlike pretty much every other area of medicine, the longer we're in it, the harder it gets.
Yeah.
So you pick any other area of medical technology, you know, drug development, whatever, the more you know and the longer time goes on, the better it gets for the patients and for you know, providers. In the antibacterial area, it keeps getting harder because, you know, as we know, as the resistance arises and it starts crossing classes and it starts, you know, becoming a challenge with every new, or virtually almost every new agent, and over time, the problem seems to get bigger rather than smaller. So you have erosion of the actual material, you know, the actual drug and the technology, and you have the broken market. So that combination makes it
very difficult,
very challenging. It's a very challenging space, yeah.
Thank you for
unfortunately.
I'd like to switch gears. Jen, can you talk a little bit about this knowledge hub?
Yeah, where's Karen? So a few years ago, we grabbed a group of, I can't remember, maybe it was 50 people, maybe a little less, for a meeting in Chicago. It was sponsored by ASM, to talk about the fact that end-to-end, so research, development, commercial, clinical use, we didn't really have an anchoring venue, let's call it. You know, there used to be ICAC, then that went away, and now what's happened is you have a meeting like this, you have a meeting like somewhere else, you know, like we've lost kind of that hub of knowledge, and critical thinking and challenging each other and just getting able to talk to people from that entire continuity from research through clinical utility, like in one room. And so we talked about this and what, you know, if we had a fantasy, what would this hub look like? Would it be an organization, a physical structure? Would it be a virtual model? Would it be a combination? What would we want to be achieving? How would we want to be communicating across those disciplines within the hub? And we sort of drafted kind of like that fantasy. What's happening now is separately from that, but also I think spurred a little bit by that exercise, ASM is reorganizing, has reorganized the entire organization. It's now scientific hubs, for lack of a better term again, rather than structured by operational units. And so AMR fits into one of those hubs. And so I'm hoping that will be the beginning of what we can create so that we have a place for very, very early stage career or people switching careers into AMR, anti-infectives, to be able to go and, find like-minded people, find experts, learn about, where meetings are happening, just kind of see everything they need, form a community, and have it be, again, end-to-end, everything from early research through clinical use, and have whatever kinds of meetings they need to have everybody come together and share just information and knowledge and support and training and mentoring. So I think that's really critically important. I think it's going to be very hard to rebuild this field without a hub of some sort.
Yeah, thanks Jennifer for that. Something I neglected to do at the beginning was just to take a poll of the audience, informal poll, just to find out their areas in which they're working. So perhaps could we just have a show of hands for those that are currently in academic research settings? Okay, a good number. And then in industry. And of those, keep your hands up, those that are in what they would consider small biotechs. So many of them stayed up. And then perhaps non-governmental organizations. Okay, thank you, that's helpful.
I would respond to this one here. My name is Robert Skov. I'm the president of ESCMID. So we have actually taken up what you've been speaking on here. So something is happening on this side of the pond as well. So we have made a strategy here 2 years ago, and where we have put in AMR as one of the strategic pillars, and have now a strategic AMR action committee who is just starting to work with the ideas that you are proposing here. But what we also are doing is at the next ESCMID Global, we will introduce Pipeline Monday, which is really focused on the early discovery, not only on antimicrobials, but also all other discoveries in vaccines, in diagnostics, and so forth. And we do have the clinicians coming there. So I think there is a possibility for ESCMID Global 2025 to have this meeting space. Thank you.
Thank you, Dr. Skov. So with that, I'd like to get on to our next speaker. That's Dr. Claudia Zampaloni. Claudia is a molecular biologist by training. She started her industrial career in 2005, working initially in biotech at Arpita and Polyfor, and afterwards then joining in pharma, joining Roche Antibiotics back in 2013, so just over 10 years ago. During those years in industry, Dr. Zampaloni served as discovery project leader and non-clinical pharmacology cross-functional expert in advanced programs, including the programs for Icloprim, Mirapavidin, as well as Nacubactam. While at Roche, she led the Zosurabalpin project, I hope I said that correctly, from discovery to the stage of entry into the clinic. And in parallel, she is currently leading the translational microbiology team and co-leads the Roche antibiotics discovery team. So it's an honor and a privilege to have Dr. Zampaloni speak with us today.
No, absolutely. I'm happy to have you here. So, hi everyone. Thank you, Erin and Greg, for the invite, for the nice introduction, and thank you to all organizers, of course. It's a pleasure to be here to talk and discuss about this section, how do we get new drugs to the clinic, and the co-speaker is Thomas and will be follow up on the same section. So I will start with an introduction about the main concept, how we see the drug discovery process, what would be important to take into consideration. And I will focus then after that to the importance of the PKPD as the main translational aspect that we have to, very critical to make sure that non-clinical then give us a drug that can move to the clinic. And I will give some example about potential consideration that we can have, challenge and learnings. And we will end with a slide for potential thoughts and potential discussion. As I mentioned, one of the key points that we may want to consider when we are in a drug discovery process, indeed, is that it has to start with the patients and it has to end with the patients. This is important, why? Because we need to define early on the target product profile. So it means that we need to have a clear idea about what is the target population, what is the indication that we want to target in this patient population, and importantly, which type of bacteria. I will not spend so much time here because Thomas will indeed, we will talk about the TPP. Then another important aspect is the choice of the modality. Of course, a predominant one is the direct acting antibacterials in the small molecule field, but in reality there are also other modalities. And the choice of the modality could be something that we can do based on what is the technology that we are more familiar with, where we believe that we can leverage our knowledge. But of course, it's a modality that has to make sense in what is our final goal, our aim. So again, it has to be something that at the end can target our population of patients. In the central part of this slide, you see what is the process itself. So our goal is indeed to find a potential, to identify some hits that we want to then develop in a potential lead molecule, a lead molecule that can be optimized with the final goal in this process of drug discovery that we can identify the clinical candidate. So the molecule that we think can go back to the target population that we identified at the beginning. This concept can be better explained more in details with the idea of define a target candidate profile. So it means that in our intention to move from identifying a lead, moving to its lead optimization, and then having a pre-IND, so a pre-investigational new drug, we need to make sure that we set very clearly since the beginning the decision criteria that will allow us to move from one phase to the next one. And we have to be very strict in the decision that we will take, because the idea would be for each of the green boxes that you see for each phase to fulfill potential criteria that we put in place. But of course, we need also to be sure that at the end, all these criteria, they make sense for what is our target clinical candidate, and that we can go to next steps. For example, in lead identification, when we want to have our initial series of hits that we can consider to move to the next step, we need to have molecules that are active in vitro, we need to have a certain level of knowledge about the pharmacokinetics of the molecule. We need to have molecules that are selective, not potentially having off-target activity. They need to be then safe. But of course, we need that these molecules, they are also also active in vivo. So a proof of concept in vivo is where to go to the next step. In lead optimization, we may want to expand our knowledge in terms of in vitro resistance of the molecule. We want then to, again, improve our understanding of the safety of the molecule, the PKPD property of the molecule, but we want also to introduce other aspects. For example, understanding the resistance. Is there any liability in resistance that we can identify already early on, and of course the PKPD. You will hear a lot about PKPD. And then when we have a molecule that is shown to be preclinically efficacious and is also safe, we can move to the next step, that is the pre-IND. Here is the stage where it's for sure very exciting. We can start having the initial indication about a potential human dose prediction. We need to increase the level. Of course, at this stage, we need to be very, very sure about the safety of the molecule, those range findings in rodent species, non-rodent, considering the GLP toxicity study, because we need to have a molecule that if it has any flag in terms of safety, it means that we have to take it very seriously and not going to move it forward. And of course also the scale up of the molecule to go to the clinic. So if we can nominate our clinical candidate, it means that we have a molecule that can go to the phase 1. So all these describe what can be, all this work is a non-clinical work, and is the non-clinical work or preclinical work that is extremely important because it helps us to build our package. It's interesting to see that, of course, in the last years now, since 2013, this is an adaptation of the publication of 2013 from John Rex. We see that indeed we are in a very privileged situation because we can reduce the number of clinical studies if there is a high unmet medical need. So if the bacteria that we want to target is a species that has a particular high level of resistance and is difficult to target, we may consider that we are in the scenario that you see here in this histogram in the option B or C. So it means that we can, in this situation, rely mainly reducing the clinical study and rely a lot on a clinical package that we define as robust non-clinical package, and it means that it's a package where we rely on non-clinical data and human PK. Of course, having a robust non-clinical package, it implies a lot of consideration, because we may ask to ourselves, okay, how much do we have to do to make it robust? So we want to consider only a classical approach in which we do in vivo studies, in thigh, in the lung, or in both of them. How many isolates we need to test? Do we want to also consider an in vitro additional approach and eventually considering dynamic model or simply time kill and then doing, for example, mechanistic modeling? Do we want to test the humanized dose? How much of the resistance data that we have we can factor in, for example, selecting our dose later on? And what is the relevance of models that are not just measuring CFU counting, but, for example, that are also measuring other endpoint to check the outcome, for example, of in a survival that is closer to what we are going also sometimes to measure in the clinic. And then, of course, also, do we want to test only the usual murine models, or we want to expand and to test also higher species? So these are all questions that we may have when we want to build our non-clinical package, but always to keep in mind that it has to be a tailored type of approach. So each drug needs to adapt the amount of work and the type of work based on what is the target product profile that we aim. So in this context, it's clear to see that in the box there, the strong or robust non-clinical package is relying on a strong PKPD. Indeed, the PKPD is really, we can define as the central pillar of the antimicrobial development, because we have to build the very deep knowledge in the PK, the pharmacokinetics and pharmacodynamic properties and assessment of our molecule non-clinically. The finding that we have, they go then together with the clinical finding from the human PK to feed, for example, what we define as probability of target attainment. So we need to combine the efficacy that we see in vitro, or let's say in vivo animals, to see how it can translate in a clinical outcome. So in the exposure that in human will translate with an efficacy for the majority of the patients that we want to target. And finally, also in phase 3, when we will be in the patients, we will have the possibility to validate that the dose that we select with the non-clinical data and the human PK, we can have a dose that indeed is translating and confirming our original hypothesis. Going a little bit more visual about what does it mean, this is again a slide showing the importance of the PKPD, and is showing the fractionation study that we need to conduct. These are conducted, we have a total daily dose that can be fractionated in once a day, 2 times a day, 3 times a day, and the data that we will get from this fractionation study will indicate us if the molecule is the efficacy of the molecule is more Cmax related or EUC related or time above MIC. We then do the dose fractionation study. Here, this type of study that are the one that you see in the middle are the one that we conduct to understand the exposure that would be important to calculate if we want to target, for example, 1 log reduction if the burden of a bacteria in the infection model that we are using, 2 log or 3 log. And then we have the MIC90 determination. This is the drug concentration that inhibits 90% of the tested bacterial isolates. So this is the minimum that we want to target with our dose. And then with the human PK, again, we combine all together, and we have the calculation of this probability that we have to cover our patients with the selected dose. So based on this background information, I choose 4 potential considerations that we may want to talk about, to discuss, but you may have also other type of questions that you are facing in your daily life when you face your drug discovery process. And the first consideration is about dissecting the PKPD driver. This is what we have, the classical textbook figures. Again, we can count on the validity of the model that are in vivo model, murine model. At the time, this picture is from Craig, so really the start of the understanding of the PKPD for the antibiotics. And very clearly we see that is a driver of efficacy, that is the time. We have more recent publication. You don't have to read what is there, simply indicating that when we have different class of antibiotics, we can have different driver of efficacy, but usually within the same class of antibiotics, the driver of efficacy is maintained. Interesting enough, in the middle of this table, you can see that not always the type of assessment that we would do, for example, with a time kill in terms of driver of efficacy would fit with what we find in vivo. And that's the reason why we need to consider sometimes both model, both in vitro and in vivo, or in vitro we may want to consider dynamic model of infection. So this is what is for a class of antibiotics that are known and are very well clinically validated. And here is the example of zosuраbalpin. It's just an example here. You will hear a presentation on Wednesday in Novel Antibiotics from Ken Bradley. But before that, is a very good example of how a novel class of antibiotics may behave compared to established class of antibiotics. This is a lung model. We see that indeed there is a good correlation for the Cmax. There is a good correlation in EUC that is just following the Cmax, but we don't have any correlation in the thigh. So we have already 2 potential drivers of efficacy. If we look now at what we observe extending the study in 4 lungs and in 6 thigh, we see that indeed the efficacy driver that we would find in the thigh is mainly the time above MIC. So again, the learning is that the PKPD relationship can be different, and in vivo model, and we may not be able to select a preferred index. And that's the reason why it's very important that early on we start understanding what are the characteristics in terms of PKPD of our molecule already during the lead optimization, and then also we consider what could be the best approach for doing our PKPD modeling. Do we use the classical PKPD index, or for example we want to consider also mechanistic modeling? The second consideration concerns the humanized exposure model. There are models that we can use in vitro, for example, chemostat or low fiber infection model, so the 2-compartment model, but we can have also models that we can apply the humanized dose in vivo. There are, of course, pros and cons in both approach, for example, the chemostat probably we use more for PKPD, the hollow fiber we would use more for resistant assessment, but also the hollow fiber, for how many days we can run in hollow fiber, when we start to consider that the suppression of resistance is good enough to consider that the dose that we are considering is the right dose. And the animal model are open to question here. Again, also they have their pro. We can consider for validating the dose, but then we have to be sure when we ask the question to this model that they are validated to answer the question that we want to really that we're asking to the model. And of course, the acute model in the mice are also used for breakpoint validation and setting. And we may want to consider then the mouse, the rabbit, or the porcine. So here is a very good example from a molecule from GSK, gepotidacin. In this case, we see that the hollow fiber infection model is working very well. Without going too much in details, if you move it from the top part to the bottom, you see that increasing concentration of the molecule in the hollow fiber when we take samples and we plate in presence of drug, multiple of the MIC concentration. There is the possibility to going in increasing concentration of the drug and completely suppress the resistant development that is on the graph that you have on the bottom right. So here is a very good example in which the hollow fiber is working well. But if we go, for example, and we check at the hollow fiber in Acinetobacter baumannii, we will see that indeed the literature, these are literature data, they show, for example, that with the except of meropenem, in reality, all the other standard of care that are available, they show to be killing in a first instance, but then to show rapidly regrowth. And we don't see the same if we test the molecule in combination or on top of another standard of care. You may appreciate the variability that we have, not always the same inoculum sites are used, and also the conditions that are used, how many days the hollow fiber is conducted can be different. And with this one, we can say that then the hollow fiber for sure is a good model, but we have also to consider that sometimes for novel mechanism of action molecule or eventually for some types of microorganism, it may not be applicable, especially for assessing the resistant development, because the efficacy is so poor that we will not have the time to assess it. And so we need to implement with additional model, in vitro model. In this table are also included the chemostat data, so it doesn't seem that the chemostat seems to be a solution in this case, and eventually considering a standing model of infection in urine or higher species. Of course, to consider that this table may be not exhaustive, but it's not that there is so much in the space for Acinetobacter baumannii. So if we consider then another consideration would be the one of which approach to take, the standard versus the risk approach. So now I just showed that we have this model where we can test, for example, a humanized dose. In this case, what we could consider, to say, okay, normally we could use, for example, humanized dose in murine model after when we are already in phase 3. But in reality, we could also consider to already test some of our dose, potential dose that we identify through the PTA. We bring back to the non-clinical models. We test the efficacy, if we can suppress resistance, what is the efficacy, and so on. And then if we see that indeed there is an advantage or a need of adjustment of the dose, we will make it and we go back to recalculate our PTA. Last but not least is the MIC. Of course, MIC has a very strong connection with the PKPD, and although it seems to be a very simple type of test, it's something that we don't have to underestimate. Specifically here is the example of zosurabulpin. Zosurabulpin early on, already at the discovery phase, was showing a trailing effect, trailing that was ameliorated through the chemistry, but was not disappearing for a large number of isolates. That's the reason why, after several testing, several conditions, several media, we identified as the horse serum 20% as the condition that is really solving the problem of the reading, and we have a clear endpoint reading that we can have thanks to the presence of the serum. The methodology has been discussed both with CLSI and UCAST. At the moment, the method, this alternative method, is accepted by CLSI and is under discussion with UCAST. All of this to say that engaging early on with key stakeholders is really the way to go, and to communicate intent and find together a solution. And there is a very high level of, they are very collaborative. And again, don't underestimate an MIC. This is an extreme case because we have the trailing and we could really recognize, but also when the MIC seems to be okay, make sure that you test different media, different supplier, that you don't have variability, because later on you need to use the MIC for quality control, for correlation with the disk and so on, and to also do your surveillance studies. And with this one, I'm the end. Some thoughts. To stimulate potential discussion. When I discussed them with Thomas, we realized indeed that some of these thoughts are indeed the part of the section that we will hear in the next days. So I'm sure that at the end of this conference, we will know much more about also artificial intelligence, for example, and machine learning, but also can we, for example, enhance our understanding of the bacteria physiology during the infection. This one could be really of help to understand, to help the translatability of what we are doing non-clinically. Using, for example, in vitro model, like for a pneumonia model, the human airway epithelial cell culture, using more, again and again, more and more complementary, of course, the mechanistic modeling, and what we can do with all the data that we collect non-clinically about resistance. Is there a way that we can really understand if some mutation, what is the evolution of the resistance? Can we do some evolutionary modeling that will tell us which type of mutation in the clinic could be the most relevant one? And with this one, thank you so much, and happy to take questions now or after Thomas' presentation.
We have a couple of minutes for questions. Thank you very much, Claudia. Wonderful talk. Perhaps just one quick question. Claudia, maybe you could provide a bit of perspective on standardization of some of the translational models. We know that there are efforts underway and have taken place already to try to standardize such things as the murine pneumonia model, thigh infection model. Are you seeing similar movement with, for example, chemostat and hollow fiber models as well?
Okay, thanks for the question. Yes, of course, the murine model are the standard one, the one that are very well validated, and that's the reason why I put the picture with the Craig, the book text there. There are, of course, many experts here, and we will hear a lot about the chemostat and the holofiber. I think that we did a lot of advancement there. There are some species that they work well, some drugs that they can perform well in the model. Having said that, of course, we need to make sure when we use them that we know what is the question, what we ask to the model, and be sure that the model is performing for what is the need of our molecule. For example, in Acinetobacter, it seems to be challenging from what we see from the literature research. Having said that, the holofiber is expected in some way. And it doesn't mean that we will not perform the holofiber. We may need to perform it, and then unless till the moment in which we will be in a situation in which it will be clear that the model can be used or not used for certain species. Maybe on the holofiber aspect, I think one effort that we can do as a community would be to make sure that we have a common approach in using it, because probably we don't have guidelines how to use it. So each of us, and I say each of the is going to approach the holofiber, say, okay, what is the inoculum? I put 10 to the 6, I put 10 to the 7, 10 to the 8, how many days? So there are many conditions that, more than one of critical conditions that we are changing. And it can be that there is not a unique solution. Some species need the lower inoculum than others. We need to be more tolerant. But if as scientific community would be together and taking a common approach would be very helpful, in general, also for other models.
Go on. Are you going to say what I'm going to say?
I don't know.
You don't know.
Probably.
You say it first then.
So just to answer that point, which is not the question I was going to ask, is I wonder whether rather than telling everyone to do the same thing, it's trying to understand the impact of inoculum and the differences we make on the experimental system so that we can choose, drug by drug and bug by bug. I don't know if that's where you were going.
No, it wasn't actually. I was just going to answer Greg's question and say that within the GNA Now program, we have got a work package which is looking at, technical factors, shall we say, in terms of both in vitro models and time kill curves, and Shampa slightly touched on that. We're not going to come out with a recommended method, but we're just going to highlight important things like inoculum, the way you prepare the inoculum, the volume that you sample out of the models, those kind of factors.
So I did want to ask you a question, though. So I'm Shampa Das from the University of Liverpool. I just wanted to touch on the building up of the mechanistic modelling, which is, I think, an exciting field. I think it can only make sense if we've got the validation from experimental models behind it. And I'm not seeing the growth of that in as much, because that's not just, doing a mouse model. What we're trying to do is, get into what's actually happening at the cell site, at the bacterial site. And I don't know, from your experience and what you've done in your area, are you seeing some more work being done on the validation part of it? So not the modelling part, but the, getting the background data.
Yeah, absolutely. As you say, to do the mechanistic modeling, of course, we need to feed the model with data. So the data that we are going to use to do the model are extremely important. And the ideal approach would be if we have more than one model that we can use in terms of data. So it means that we use in vitro data. That, of course, is the potentially where we want to go, no? Reducing the animals type of experiment, having more in vitro. But of course, this means to use the in vitro data, could be a static model, could be the dynamic model, like for example, chemostat or holofiber, plus the mouse model, lung model, Thai model, and then see how the model, they correlate, what does it mean, how do we need to adjust our modeling to make it work. But of course, after this initial validation, then it means that we should be able to use the mechanistic modeling, because the model is supposed to be applicable to the understanding if our drug can work or not. Of course, it's also possible to have a different application of the modeling, in the sense that when we have, for example, difficulties in extrapolating which one is our best predictor of the efficacy, is the Cmax, AUC, or the time, the mechanistic modeling could be really the one that could help to see if we are more in one direction or another one. So it's an additional, of course, at the moment we are in a situation in which we cannot work without the classical approach, but this approach needs to be more and more taken into consideration.
more for the non-traditionals, you know, where the classic approaches won't work. I think we need more than just the hollow fibre and the animal models, and I think we need to understand, you know, again, things at a cellular level, what's going into the cell to really feed into those models. And I'm just not seeing that methodology out there. It'd be good to see that published.
Thanks.
We'll just take time for one more question before getting on to the next speaker.
Very quick question. Thank you very much. I'm Diana Aguilar from the University of Zaragoza. I'm a researcher that we just implemented hollow fiber system in our laboratory for anti-tuberculous drugs. And it took us a long time, like 3 years, to get it really high and now recognized by the European Union. The thing is that on the process, we realized that was a very challenging to deal with molecules because some compounds are not compatible with fibers molecules. How are you doing with that?
Yeah, that's a very good point. Indeed, the starting would be to test the cartridge, the different cartridges that you have with the molecule and to see which one would be the best fit. Based on the paper already, the characteristics that are provided by the supplier as well as the chemist, you should have already an indication if a type of fiber would work better than another one, but the best would be really to try to do your PK and then bioanalytic to see what you get. If what you go in is also what you get out from the system. So real life experience.
Thank you. And maybe we could move on to the next speaker, and you can pose your question after.
Okay.
Thank you, Claudia. And our next speaker is Dr. Thomas Doyle. Thomas, unfortunately, was stricken with COVID just recently, so he'll be presenting virtually from not too far away. And he's going to be covering, essentially piggybacking off of Claudia's presentation, bringing new drugs to the clinic. So Dr. Doyle is a physician scientist who is trained at University of Cambridge as well as University College London. He is a certified medical virologist, and he joined GSK in 2018 as early development leader on non-traditional agents primarily. And currently he's the vaccine development lead at GSK in their vaccine and ID research unit. So it's a pleasure to have you here, Thomas. We're sorry that you can't attend in person, and all our best for a smooth recovery.
Thanks very much, Greg. Can I just check that you can hear me okay? Okay, please, if someone can send me a message if they can't hear. Thanks very much to the organizers for inviting me to speak, and sorry I couldn't be at the auditorium in person. Here are my disclosures. So I may have missed some earlier comments on the TPP, but I wanted to start by spending a few minutes talking about the target product profile. So the target product profile, sorry, I'm getting a lot of feedback. Is there any way I can correct it? Okay, if not, I'll try and continue. So the target product profile is the document that lays out the fundamental characteristics of our product that we're trying. it may influence the comparators that we use in these studies, in addition, or additional agents we use. And in general, what DDIs are acceptable and the toxicity profile that's acceptable for particular molecules does really depend on which population that they're going into. There are different considerations in older and younger populations, as there are between men and women potentially, and maybe different considerations if we're going into a severe indication versus non-severe. And some broader general points on TPPs. I think it's useful to have early physician and clinical input. So that's to make sure that we're using contemporary medical categories and patient pathways, and making sure that we're acknowledging standard of care and where we fit in relation to it. And I think it's also helpful to move towards medical diagnostic language in our TPPs, particularly when we're talking about organisms or categories of organisms. Just to point out that innovative TPPs aren't easy, and they may need to develop and become more granular over time. We may need to develop our epidemiological data and other types of data to support their development. We want to make sure that we apply any available regulatory guidance that pertains to the indication or to the organism, and to be mindful of timeframes in the diagnostic pathway, so contemporary hospital or clinical diagnostics. For instance, if we need an organism ID, we should be mindful that this will become significantly after presentation and potentially antibiotic admin if we need to identify that organism before we administer our drug. We should always look for differentiation in our TPPs, and a practical consideration is that we'll be making sure that the value of the product matches the aspirations of our organization. Okay, so we thought it'd be a good idea to lay out the typical matrix team that would be involved in the discovery phase, maybe approaching an IND or initial CTA. This is always dangerous to do because there's the likelihood that we'll leave off some important functions, but I'll give it a go. On the lower left-hand side, Claudia had talked about the interaction between biology, microbiology, and pharmacology in the early phases of antibiotic discovery. Obviously, our chemistry biopharm leads will be fundamentally important during the early design of our agents. Our CMC team will take over the development of drug substance and drug product into clinic, and will plan manufacturing campaigns and study supply. This needs to be planned years in advance, so appropriate that we get this right. Our IP attorney will submit our patent applications and protect our intellectual property. If we need to work with CROs, which most of us do, we need to choose quality CROs and maintain a good relationship with them. Our toxicology team will help us to identify and characterize any toxicity associated with our compounds and help us understand the therapeutic index. And we'll do extensive PK and metabolism work, both in discovery and moving into early development. More on the right, on the clinical side, up top, we have our clinical scientists and clinical safety, designing our studies, designing our clinical development plan out to launch. Our clinical operations lines will help us to implement and deliver clinical studies. Clinical pharmacologists will help us translate our PKPD models into humans. Many of us will be working with funders who we will apply to and report to potentially. We don't want to work in a bubble, so external expert consultation is important, both medical and scientific, to make sure that we validate our internal plans. And our regulatory colleagues will obviously be crucial in pulling together the initial IND application and making sure we make the best use of our pre-IND meeting. And even at this stage of development, most of us will have some sort of input from our late phase functions, such as medical affairs and commercial. So as Claudia said, we want to talk a little bit further about modalities. Lots to talk about here. So this is just a simple slide acknowledging that traditional antibiotics are generally thought of as small molecules that kill or inhibit the growth of bacteria. But as we know, over half the programs in the antibacterial discovery pipeline are non-traditional agents, that is something else. That doesn't include vaccines, but I've put vaccines up there because they're still an important part of the control of bacterial disease, and there's much ongoing work on vaccines against AMR pathogens. The COVID-19 pandemic has somewhat normalized the use of antibodies for treatment and prophylaxis of infectious diseases. And in the antibacterial space, of course, we have the licensing of bazitoximab for the prophylaxis of recurrent C. difficile infection. But these are also very flexible modalities. We can make modifications to the Fc domain to increase, decrease half-life, further modifications to facilitate, defacilitate interactions with various arms of the adaptive immune system. So I think a lot of interesting work to come from antibodies in the next 5 to 10 years. Over further on the bottom left, I've represented the anti-adhesin approach, of which the GSK Phage antagonist is a good example. The strategy here is to block adhesion of bacteria to epithelial cells or other surfaces. It's also classified as an antivirulence therapeutic, of which there are many other examples. And the idea here is that we look at something that affects, that relates to bacterial pathogenicity, but not necessarily parts of the bacteria that are required for fundamentally for its survival. And less familiar to me, but equally important, are bacteriophage strategies. So these are very interesting agents that can infect and lyse bacteria and be designed to deliver different types of payload to bacterial cells. There's been a lot of interesting reports of successes in bacteriophage treating patients with difficult to treat AMR bugs. And I think we await further randomized studies to fully understand how to design, implement, and use those strategies. The COVID-19 pandemic has also dramatically accelerated the interest in nucleic acid-based therapies, both therapeutics as well as vaccines. And there's much ongoing work on human targets and virological targets. And there are some challenges with developing these for bacteria, but a lot of discovery groups working on targeting RNA-based modalities in particular to bacteria to try to look at mechanisms for getting them inside the bacterial cell. So moving forward then, what are the potential benefits of non-traditional approaches? Well, these are new classes of antibacterials, and so in that sense, they invest in the urgently needed innovation for AMR. It's possible that some of these targets may not induce the same strong selection pressure in the bacterium, so that may have implications for resistance. Toxins could be an example of that. Because some of them are microbiome sparing, we hope that they won't facilitate the accumulation of self and cross-resistance in the gut, like traditional antibiotics. And as we've seen, they can have different durations of activity and protection, which is great for increasing the versatility of our pipeline. Some potential risks that people see, there could be significant differences in the probability of success. As we mentioned earlier on, if we get good molecules into phase 1, then a lot of our antiviral, antibacterial agents have high probabilities of success. There's, I guess, the issue here, because we're not directly killing or inhibiting the growth of bacteria, we may pick a redundant target or a target that's not actually on the molecular pathway of disease. The animal models will need to be new and innovative. Some agents may require a companion diagnostic if they're organism specific. And in general, the interrogation for mechanisms of resistance may be more difficult, and we might end up not fully understanding some potential for resistance until we get further along the pathway than we would usually be. And the last few points I'm going to talk about in a little more detail just on the next slide. So one of the challenges is the absence of the observation of minimum inhibitory concentrations, which of course we don't see for many of these agents. And while this is something we can likely get around in the long term, it does affect the way that we develop antibacterials, because a lot of our microbiology packages, etc., are centered on this observation. Good examples are spectrum of activity studies. How do we want to do them using multiple different organisms? Susceptibility and resistance determination in clinic. Our surveillance studies pre and post-approval. But other approaches here have significant potential. We have binding assays, depending on the mechanism of action, other functional assays such as neutralization assays, cytotoxicity, invasion. But I think more broadly, genotypic and sequencing techniques will probably come to the forefront as they are for some other types of infections, and helping us to understand resistance and other aspects of these non-traditional agents. As we've mentioned, PKPD and human dose prediction can be really quite challenging, initially at least. That's because we need to apply innovative models where we don't have the same experience or the same translational predictions. And we need to build maybe specific models for organ systems, further understand the use of really the microbiological and survival thresholds that translate to clinic. If you're interested in this a little bit for the FIMH antagonist, then Jennifer Hoover is going to speak briefly about an approach in FIMH antagonists on Friday. And also for modalities like antibodies that we might expect to use in combination, it would be important to understand, how we should approach this for PKPD, what the rules are for single antibody versus combinations, both in the preclinical phase and in the clinical phases. Another item that comes up a lot in the literature and these discussions is the position relative to antibiotics. I guess for a lot of us, for serious systemic infections, we think that most new therapies would have to be used in addition to antibiotics and not necessarily head-to-head. And this creates some difficulty in that you would need to demonstrate superiority to antibiotics, therefore, on traditional regulatory endpoints to get registered. And this may be difficult to achieve. So we probably need ongoing interactions with regulators to understand what endpoints would be acceptable or what other approaches we could use in our pivotal studies. So I wanted to finish by talking a little bit about the FIMH antagonist. In 2011, the Holcomb Laboratory demonstrated that inhibition of E. coli infection by FIMH antagonists in mouse cystitis models. And then in 2016, GSK and Fimbrion entered into a collaboration to develop FIMH antagonists for uncomplicated UTI. These antagonists are not traditional antibiotics. They don't kill or inhibit bacterial growth, but they inhibit binding of FIMH to glycoproteins on the bladder epithelium. They thereby inhibit attachment and invasion thought to be required to sustain UTI. Apart from E. coli, there are other bacterial families that contain FIMH, so Klebsiella, Enterobacter, Citrobacter. But for these organisms, the role of FIMH in the establishment of UTI is less clear. We did bring FIMH into early clinical studies, and on the left-hand side is our first time in human study, which was supported by CARB-X. And it was a typical single ascending dose, multiple ascending dose study, looking at safety, tolerability, and PK in healthy volunteers. And we concentrated on the PK in the urine because that's the site of action of the drug, and making sure we got past urine thresholds that we'd establish in the animal models as potential targets for efficacy. And on the right-hand side is our phase 1b study, which is currently ongoing. So this is a study in women with acute uncomplicated urinary tract infection, where we're looking at safety, tolerability, and PK. But the primary endpoint is microbiological response to the FIMH antagonist in urine as a proof of principle. And the general scheme there is that we recruit women with uncomplicated urinary tract infection. We randomize them to 5 days of either the FIMH antagonist or nitrofurantoin. And then we look at the microbiological outcomes at the test of cure. So that's day 10 to 13. And in addition to those endpoints, we'll have exploratory endpoints that I think are generally interesting to the antivirulence field. So looking at microbiome studies to see if we have the microbiome sparing effect compared to antibiotics that we expect, and also a preliminary look at sort of resistance potential. So thanks very much, and I'll hand back over to the organizers. I can't hear. No.
Mandar alguém para dentro de sala. Ou então tu agarra o teu telefone, faz esse instante. Hã? Não, é sempre a mesma reunião. Então vá, bora lá. Não, eu preciso é que tu entres. Eu vou ter que pôr o som para dentro de sala ao mesmo tempo que eu estou a falar. Que é para ver se não faz o bo. Mas se calhar depois se quiseres só colocar no PA, só para ver se... Então bora lá. Também está cá. Agora preciso de enviar para dentro de sala, ao mesmo tempo, que é para... O meu, este micro... O Zoom e... Não. Preciso estar com os dois áudios ao mesmo tempo dentro de sala. O Zoom e os microfones. As pessoas têm que ser ao mesmo de sala. Ou seja, tem que simular como se fosse um sonicamente. Bora lá. Tirar a música. É para a sala, claro. Não, eu quero este microfone dentro de sala com o áudio dele. Desculpa, com o áudio dele. Agora metes o zoom da sala. Zoom da sala. Ok. Fala lá, Nuno. Alô, alô, alô. Fala, Nuno. Vai só lá fora mesmo falar assim. Olá a todos, eu sei que vai ser muito alto. Só para a gente ver se realmente a coisa está a dar ou não. Fala mesmo alto para a gente perceber se isto está a fazer retorno ou não. Fala lá, Nuno. Estás-me a ouvir bem? Hein? Ok, corta.
Good afternoon. Welcome back. That was a wonderful earlier session of talks, and we're looking forward to closing out this session with another set of 3 talks by industry and regulatory experts. Just a quick reminder that keep your questions in mind, written down. If you prefer to write down your question, Carla and Chiara have circulated some question cards that you could submit back to them, and we'll try to get to as many of them as we can during the roundtable. Thank you. So I'd like to introduce our next speaker, Judith Steenbergen. Dr. Steenbergen has over 20 years experience supporting antibiotic development programs, both at small biotechnology companies, including Cubist and Paratech Pharmaceuticals, and also has extensive experience in both pre-commercial and commercial stage settings, with a strong background in scientific and medical affairs. She is president and founder of Scientific and Medical Affairs Consulting, and she provides strategic scientific and medical affairs consulting to small biotechnology companies, specializing in global scientific strategies, as well as including scientific platform development and gap analysis. So with that experience in mind, we're glad to have Judith speaking with us today. Judith.
Thank you, Greg. All right, and thank you for allowing me to speak today. Figured, based on the previous presentations that I heard, I'm actually going to do a little bit more in-depth background on who I am and what I'm doing and why I'm still doing it. Hopefully giving some people a spark of hope that there is a potential future. Things will have to change. I think we all agree to that, and there's going to be a lot of presentations this week on AMR, different issues within regulatory development and all those things, but I do think that there is, obviously there's still passion and there's still a need, so that's what drives me. So yes, I have a PhD in microbiology. I did my PhD work actually in virulence factors and new model development, so it was animal model development, et cetera. I never once went into a lab when I joined industry, so there is no background there for me anymore. And I have to say, which I'm going to be very honest now, I've never run an MIC. So my background is very different. I joined Cubist Pharmaceuticals in their clinical microbiology group, and I was there for about 13 years as a microbiologist, working on the translation. So love the earlier talk we had on discovery and all of the things that go into that. Really what we would do is once there was a drug that was discovered, had met the go, no-go criteria, is what do we need to do to develop this from a really specifically a microbiology perspective. So we would sit on the clinical development teams, work on clinical trial design, be part of the data analysis teams, statistical analysis plans, all of those things that are needed to bring things forward. We're a team with our clinicians, our MDs, our statisticians, our research associates, et cetera. So that is what I did. I've been incredibly lucky to have brought multiple drugs to the market with these amazing teams in mind. So everything from daptomycin, which was the first one I was lucky enough to fall into, through the other compounds that Cubist did develop. At the acquisition of Merck, I did not stay on at Merck, so I do not have large pharma experience, which I do think what Jennifer said is very real. At large pharma, there are many more opportunities to have other positions, other jobs, other roles. I was pretty attached to my clinical micro background, my expertise that I was able to gain within that, and I loved clinical development, and I knew antibiotics. So moved on to Paratech after that and was able to get both their rhamatocycline and the sarecycline, worked with the teams on those to get those across the finish line. What I did get at Paratech is that they said, well, you really do know the space, you know the people in the space, why don't you do a little bit of medical affairs, you know science strategy, you know publications. So at Paratech I was able to start and build out a medical affairs team, which was, I think, an amazing opportunity. I left Paratech and started SMAC, as we call it, for short. And I've been really lucky from a very different perspective now to work with, I'd say, over 20 small biotech and pharmaceutical companies to help work with them to design what types of studies they need to do, how are they going to fill the gaps, but with the end in mind. And we're going to talk about that within this presentation. So my request was to talk about IND to NDA. And I'm not going to speak that much about IND to NDA. I'm actually going to speak a little bit about what that looks like, but more about what can we do within that time frame that's different, because there's pretty prescribed information that's needed from IND to NDA. So we're going to explore that together. So here we go. I did like when we were having our little discussions earlier on that there's a lot of academic individuals here, and and maybe it's because I'm older, I do see some younger faces in the audience, so I do think this is a really nice way for them to see a lot about what's needed preclinically, what do we do, and really what does it take to get these antibiotics or anti-infectives as more of a general topic to the market. So again, you've got your preclinical, which you had a great talk about already, and then you get to go into phase 1, which would be your first in human study. In the US, let me preface this as well. I have filed INDs, NDAs, as well as MAAs. However, this will likely be a very US focused because that is primarily where my brain has been at. So I apologize for that. But yes, so IND, first in human, and we can talk a little bit about that because that's not always necessary depending where you're at. Phase 2, more people. Phase 3, even more people. You get to file your NDA, MAA, and then you're moving on to market. So we'll get into more details on all of those steps. So when they asked me to talk about it, I said, well, what am I going to talk about? Everything's in these guidance documents, right? And these are regulated events, so you have to follow the regulations. We've got everything from what you need in your microbiology sections to what you need, how you need to put the document together, the formatting. The first one I was ever on, which was the daptomycin one, they basically looked at me and they said, oh, the TOC and 267 and 4, and then we need to do this, and who's doing that? I said, excuse me? Where do we start at the beginning? It is a very massive compilation of data that goes into these documents, but they're all prescribed. They're in these regulatory guidance documents, and if you haven't looked at them, if it's specific things you're interested in, look, right? They're there. But they are very specific. And I think what we're going to hear throughout this conference as well is that that may not always fit what we're trying to do differently today. So we are going to have to start working on, how does that push and pull, not from finance, I'm sure that's a whole different problem we have, but what needs to change so that we are developing things which we know patients need that make sense, but that also are able to get approval. So we're going to look through that as well. So we're going to talk a little bit more, and for those of you who've been in this space a long time, this is going to be very basic, and I'm not going to give any specific examples, but happy to talk to anybody about it. The IND is your preclinical research. This is all of the lab and animal data. Currently, again, there's different ways to do it, but right now MICs are a big way that we are showing potency. This is also your early animal efficacy models in vivo, looking at your early safety. This will be all your tox work that has to get done as well in animals. You can file an IND submission that goes with it. That's a big regulated scenario. And then you can do phase 1 studies prior to IND outside the US. So there are many companies that will do their phase 1 studies outside the US and then include those phase 1 data in their IND. In the US, you cannot run a study in humans without an IND. So then we move on to the clinical development phases. Super simple, right? Phase 2, 3, and then you get your regulatory review. And again, these are outlined. Phase 2, you're going to do your efficacy, see high-level side effects in a larger group of patients. High-level efficacy, it is not going to likely be powered, especially in anti-infectives, for anything to show efficacy, but you should see maybe trends. And then phase 3 is your confirmation, statistically powered, your endpoints are well-defined, and you're going to follow the guidance documents on that. Then you compile all this great data, send it off to the agencies, and you either get your label, your package insert, your marketing authorization, all of this gets done, and you're able to move forward. So, great. I will go into a little bit more, and this is a very, very selfish perspective. This is the microbiology side of drug development, primarily. Where does the microbiology data, which also includes animal data, PKPD data, et cetera, more of that type of data set. Where does that go? Where does that play out? And this is really over time, how we try to play into all of these different phases. And I know I see people taking pictures. I'm sure I'm happy to provide this or to be provided the slides because it is a bit small. But there's a lot of various parts. And what you'll see on here is under discovery is your TPP, which we talked about early in today as well, your target product profile. I know Claudia also brought it up. I'm actually going to spend a little bit of time at the end on our TPP. I think it is one of the most valuable things that a company needs to put together, and they need to get alignment and understand really what you're developing, who you're developing it for, and does it really make sense to develop it? And that's hard, right? We're passionate about the programs we do. We are passionate about the things we're doing. The reason we're in anti-infectives is we can save lives, right? It's a pretty cool thing to say. So it's hard to say no-go to those programs. So another part that goes into this is really what your susceptibility testing strategy looks like. I'm not going to spend time on that either, but the susceptibility testing is quite important. We talk about diagnostics. We also talk about the right patient. How are we going to identify that? And currently, there's been a lot of change in the attempt to push things different directions for susceptibility testing, but we still start with the basics, which is this MIC testing, which will not work for most of these non-traditionals. So how is that all going to play out? So there's clearly a role for people to do this work. And I forgot to say in the beginning, because this was also brought up, I have been able to feed my family on my salary for 20 years. So I may have not have won the lottery and had the big house and all of that. I've actually been pretty lucky. 2 kids in college, all of that stuff has worked out and with doing the passion that I have. Okay. So objectives. Your objectives in the different phases, this is me saying what the objectives are. Obviously, there can be slight nuances to this, but really your preclinical is let's find us a good compound that meets the criteria of what we're looking at, right? What pathogen we're looking at, what are we interested in, what drug resistance are we interested in? That's the objective. Your phase 1 is it in healthy humans? Is it safe, tolerable, and what's the PK look like? Is this actually going to be druggable in people? Can we get the concentrations, what we think we need to, or range therefore of, in the right compartments in patients, in humans? It's not patients yet. And then theoretically, that gives us a dose range to start with. Phase 1 studies, which are also not necessarily, so your SAD, so your single ascending dose, and your MAD, your multi-ascending dose studies are done as phase 1 studies, but there's other ones that are done later while you've gone into phase 2 and phase 3 to supplement your NDA to make sure that you're collecting data for the right patient populations. So this is not comprehensive, but you'll be able to look at renal impairment patients, hepatic impairment, BAL studies. So what concentrations are we getting into the lungs? And now it is expected that you know that you get lung concentrations before you start any pneumonia trial, which would be logical. Wasn't always the case, just so you know. So the phase 2, you're actually going to start looking at efficacy. Again, it's not going to be necessarily powered in any way for that, but it is going to, you want to make sure that it is working on the target and the pathogen, and you're decreasing the pathogen. So these will be infected patients. And you're going to evaluate the safety as well. And this is when you should determine your dose. Right? So what dose will you be using? So there are different indications, and that's also part of the guidance documents. You'll be able to take a look that there are many indications that have guidance documents. So you've got your community-acquired bacterial pneumonia, you have your HAP-VAP studies, complicated UTI, complicated intra-abdominal infection studies. So those have existing guidance documents that you would look at, and you're going to work towards filling those holes there. So phase 3 is the big study, right? It's the big study. Used to be, you do multiple big studies, you still companies are doing larger studies still, but you're confirming actual efficacy and safety. And this is what will then be really the last thing that should make up your NDA compilation of things. Okay. So now, within that same format, It's really looking description of the things that happen. And someone talked to me right outside earlier and says, well, clearly we're CMC in all of this. It is the most expensive and highest risk, and when are we making these decisions? And he's smiling happily that he gets mentioned. But yes, I am not going to talk about CMC. CMC is a whole big black box to me that I'm always really happy when it works. And often it has been the thing that has delayed an NDA for me. So CMC is important, and formulation, therefore, is also important. You're going to have an oral, you're going to have an IV. This morning they talked about transdermal. There's all these different ways to potentially have to dose drugs to get the concentrations you need. So you need your serious chemistry and formulation, CMC team to be involved as well. And then that, maybe one day we'll have a talk about that, because that is a gigantic amount of work that goes into these CMC packages, because they have to show and prove and do and make big batches and label and all that good stuff. Meanwhile, supplying your clinical trial sites, and you can't run out. So phase 1, your patient population, as I mentioned before, is your healthy volunteers. Your major endpoints are safety and pharmacokinetics in general. Your phase 2, you're looking at actual patients within theoretically the indication that you're going to study, though sometimes you look at a different indication and you move to a new indication for phase 3. That happens. Your major endpoints will be efficacy and safety, primarily safety. You're really looking to find if that dose is going to be safe, and obviously you need it to be within the effective range as well. And then your phase 3, this is your primary efficacy endpoint, right? This is, does it work the way it's intended to work in the patient population that you're studying? Yeah. So we collect a lot of data, and I'm going to show you a little diagram in a minute about the insane amount of data. But yes, we collect a lot of data, and I think many of you are involved in various different points within this process, if not all the points. But even when you're looking at your preclinical data, you're making the reports, you're generating data, you've got to, there's specific rules around the best way to manage that, and you can't forget you have the data. So when you do file all of these documents along the way, all of that data comes along with it. And no matter, the more experiments you do, the more times you can find something that maybe doesn't fit exactly the way that you anticipated, which then results in additional studies, right, and looking at more information. And then same with your phase 1 clinical trials, CSRs, so clinical study reports are big, but everything that goes behind them is big. Your PK reports, your modeling that, you'll have PKPD modeling, probability of target attainment modeling, you're going to do pop PK. All of these things are big reports, big data sets. Everything has to get compiled together. And all of that goes into your dossier. So this is the document that contains all of the technical data. So that's administrative, quality, non-clinical, clinical, and the pharmaceutical product information, as well as your CMC. It includes everything. And they made this really pretty picture, which makes it look simple, right? This is what all you need. You just have 5 little modules, and there you go. Don't know 100%, but I know when we filed daptomycin at Cubist, which was 2003, so just last year. And when we filed that, it was one of the early CTD options. And we hadn't worked out the kinks with the FDA, and nobody had worked out the kinks exactly right. But when we ended up doing this, we had these, we still sent the truckloads of paper. So we sent actual truckloads of paper dossiers to the agency and electronically. Then once we sent the electronic, we realized systems didn't align, so then we sent a human to go help the FDA make sure that we were all looking at the electronics and the links and everything. It was a massive production of an insane amount of data that went to the agency. Harder to see that when you're doing it electronically, because you're generally in your little piece and you're signing off on what you're signing off, but these are massive amounts of data that's going, and everything is linked and appropriate, and you do your best. We've always do our best to make it perfect, but it never is. There's always going to be questions, and there's always going to be a link or something that didn't quite go. But the big modules are, you've got your administrative information and your prescribing information, and we're going to talk about the prescribing information, because that's my favorite. And then your common technical document summaries. So this is your clinical, your safety. This will be your microbiology, summary sections, all of those data that goes in there. For me, selfishly, it's outcome by MIC, outcome by pathogen, all of that will go into these different sections. And as I said, they have all very specific numbers, and this is in the guidance documents. And you're going to fill in the pieces for all of these things. If you've never done one, look at an IND before you look at an NDA. It's a little less overwhelming. Quality, clearly important. Non-clinical study reports, this is all of those reports you've worked on all of those years, making sure someone has signed them, that they're all ready to go, and they're loaded up in order so that they're referenceable and there. And then your clinical study reports. So what happened in your clinical studies? I keep looking at the regulators. I'm sure they're going to find things wrong with my way of interpreting. But so then the goal is labeling, right? What do you get at the end of this? You want an approved product. An approved product has a package insert, a label, however you want to talk it, and that's the goal. That's what we work towards. But it's also currently not, well, we'll look at it real quick. So I tried to be non-offensive, and I picked Avicaz. I figured no one's working on that today. So, so this is the label from Avicaz, and I highlighted my microbiology-centric information in here. But again, so they studied it for intra-abdominal infections, and they studied it for urinary tract infections, including pyelonephritis, as you see here. And if you've never looked at a package insert for an antimicrobial, spend time doing that, and spend time looking at different ones. Like I said, this is my favorite part. Because this is the end, this is everything you've done, but how can you truthfully differentiate yourself from some other compound? And that becomes very difficult in a label, but we'll talk a little bit about that. But yes, so here you can see in your indication, you have your indication, so what your actual infection you're treating is, and the pathogens that you saw in the study. This isn't the pathogens you have activity against only. It doesn't include everything. It includes what you actually saw in the study at enough of a number where you could guess that you had efficacy against these pathogens. Plus, you have all of that preclinical data that goes behind it, right? This is not just, oh, I saw 10 Pseudomonas, so now we can say it has Pseudomonas activity. You clearly have to have all of that preclinical data to back that up. But this is how you get your pathogen information, okay? But where is AMR? We're talking a lot about AMR. We all know AMR is a problem. We've all, I think, in this room, we're all aligned on that. But where is that in the package insert? Well, it's not really evident. It's not big in the package insert, which it becomes a problem. So how do we differentiate, and where do we differentiate? And there are some spots now, and there's been new information and new guidance that you can speak as sales organizations can now speak as long as it's within what the label's intended. It doesn't have to be exactly on the label, so there's been some change there. But if you don't know where this drug is different from where Avicaz is different from ceftolozane tazobactam, because the label doesn't tell you, it becomes very hard for understanding how you would use it, when would you use it, so why would I use it, right? It's a very complicated conversation to try to have with individuals who do not live this the way we do day to day. So where can we differentiate? So as I mentioned before, the big clin microsection in all of these documents is 2724. It's called Special Studies. It doesn't say microbiology, it just says special. And in there, we do our clinical study microbiology data, so exactly what you got in your clinical studies. So those are the pathogens, the MICs, and a molecular characterization. All of that goes in there. We have our resistance data. So this is preclinical data. Clinical data as well, if you have it, but preclinical data, resistance incidence, how single-step mutation versus serial passage. Now there's a lot of hollow fiber passaging as well. We do a lot of resistance work to, one, understand what the mechanism of resistance is, how quickly it develops, what are the implications. We look for cross-resistance with other agents. We look for antagonism, synergism with other agents. There's all of those things that go into this package. And then your in vivo microbiology studies, so your animal efficacy, your neutropenic mouse thigh, your lung infection models, looking at isolates with resistance to other agents. Did it work? Did it not work? It's a lot you can do within this section, but this does not end up on your label particularly. But it is important, should be in your publication plan, and should be 100% part of your TPP and why you're studying this drug, right? And then we look at our pharmacokinetics and pharmacodynamics, which we've already heard about, so there will be no additional information from me, and we have many experts here on that. We do quality control, susceptibility testing, how do you test, when do you test, what makes you, how do you have to test differently, and it's very important that you include your quality control data from your clinical study as well in these. And then clinical trial information. What was the study design as it relates to microbiology? Because there's a whole clinical section that happens. But what was your study design? What were your protocols? What did you do in your central lab? The additional testing? Did you see any differences with MICs or molecular characterization of your isolates, et cetera? And so then we do have a section in the label. It's called the microbiological information, and we'll look at that. So we do actually get a little bit of a win here, and that's changed over the years. I can tell you the early years were very different than what I'm seeing now and what we're able to put into these labels, because it is very complicated, right? And there's reasons regulatory agencies had not put it in the beginning. MRSA was allowed to be in a label when I did daptomycin, and VRE was allowed to be in a label, but there was no other resistance in your indication section that could be in there because it wasn't all the same molecular subtype, et cetera. So things have grown and changed. So in the clinical pharmacology section, so section 12 of the package insert, you've got your mechanism of action, which is not what we would say is a mechanism of action, because every one of them will say it's an antifungal, it's an antibacterial drug. Currently, that is the mechanism of action. But pharmacodynamics, we do get to include in this section what the driver is, which is helpful when it comes to understanding dosing. It's also helpful when it comes to understanding the studies that we've done. We also, in our specific microbiology mechanism of action section, it includes what the actual mechanism of action is, which is important to identify. And then within that section, we now have and again, this is an older one. So Avicaz is much older than many of the new ones coming out. But I wanted to show this was kind of a big turning point, I'd say, for the ESBL space. They were able to include things along the lines of which ESBLs they'd shown activity against, which is a big deal, right? If you're going to be developing something against an ESBL, if it can't be in the label where it's going to work, it is very hard for someone to explain that to you and to understand how to differentiate. So this is here. Resistance section is generally, no resistance with no cross resistance, or if there is, it will be defined here. Interactions with other antimicrobials. Want to confirm there's no antagonism. There's not usually a lot written about synergism because it tends to be very specific, but you want to be able to say that. And then, I think the bottom section here was avibactam restored activity of ceftazidime in animal models as well, right? So these are all important pieces, and this was, I'd say, a pretty big deal when I saw this label come out. They did a really good job of trying to incorporate all of this data they'd used before, and this label also came out when you could only speak to what was on the label. So this was pretty, I'd say, pretty big deal back in the day. Then, and I didn't highlight this because the whole thing would have been highlighted, then you get your list of actual organisms you saw in the clinical trials. And then there's a secondary pathogen list, which I'm not going to show you, which is other species that you've shown activity with 90% of isolates appear to be susceptible based on where you're at. So that primary and secondary lists are quite important as well because it gives people kind of an understanding of a spectrum. If you run a clinical study, you may not see all the pathogens you have activity against in enough numbers where they would make your label. So it's nice to have that secondary list to give physicians an understanding of where the activity comes from. So that's it on labeling. But this is the goal, right? The goal is to get a drug. The real goal, though, is to make money. Let's be clear. And that's where we're all hurting. And sure, there's going to be a lot of discussions about that later. I will not be talking about that. So challenges, right? We've got scientific and technical challenges. There's resistance. Resistance increases over time. It doesn't decrease over time. Different mechanisms from one drug may affect another drug. We have to understand that. There's complexity in identifying a target, difficulty in finding novel targets or novel agents. And we've already talked a little bit about these non-traditionals that are being worked on. Those definitely will be a scientific and technical challenge. What is the data that we're going to generate? What data is important? We can't use MIC for many of these. The animal models may not work for many of these in the same way. So how are we going to identify that? What are our scientific and technical challenges there? And it's expensive. You're looking for a long time to find the right thing to get it even to your first in man, right? Regulatory, currently our indications are reasonably limited. And there's a reason for that. There's a number of infections, but we need infections caused by a specific pathogen, though now we can do pathogen-specific trials. Solder did a pathogen-specific trial. So, there are options to do things slightly differently now, but indications are limited. We still do primarily non-inferiority trials, and that's a numbers game, and both financially as well as patients and identifying all of those things. So we result ourselves in these limited labels. This is really where I think we land is, when is it worth developing a drug then? If we can't differentiate on the way that we're doing, what are we going to do to be able to differentiate it so that we have a viable drug candidate? So market challenges, again, there are much smarter people who will be talking about all of those market challenges at this particular conference with all of you. But I think we all have heard the things that are out there, right? There's a limited market out there for antibiotics. There's a lot of money currently being put into, developing drugs. I think that's been very much improved on, but now the market hasn't changed, and we make drugs to put on the shelf, which is kind of a problem when it comes to making money at the end of the day. And as Jennifer mentioned at the beginning of the talk, people want to pay out. People do want to get these noticeable increments in pay, et cetera, and they want to see a career future within that. So I do think this is a big issue for us. And then pipeline and innovation gaps. We've got the brain drain, as we've talked about it, but we also so there's decrease in research that's going on. And then there's people are trying to work between these complicated things, right? Can we make a new class? Can we do something different? But are we going to be able to get an approvable drug at the end of the day? And so those are complicated. So what is working? I mean, again, I'm going to always be a little bit positive because I'm going to continue to be in this field, and I like it. So innovative research approaches, I think people are doing some really interesting things. And again, that was just previously discussed, so I shouldn't even discuss it because he did an awesome job discussing it. But people are absolutely coming up with new types of agents. They are coming up with new ways of looking at things. There has been, as I mentioned, better funding, ways for people to be able to get these drugs developed, which is really amazing. We've seen regulatory improvements as well, right? There, as I even mentioned, now even discussing things that are not on the label but are, at least consistent with the label makes a difference. Seeing fast-track designations, accelerated approval pathways, different ways of trying to make it a little bit easier and better for these microbials to be developed. And as I mentioned, there has been increased funding and incentives. So that, I think, is what is working. Strategies for improvement. And I actually am going to end up partially blaming all of us for our problems as well, just so you know. But so we need to, enhancing research and development. We need to continue to put money into basic research. We need to have people who are interested in basic research. It is absolutely vital to have innovative treatments at the end of the day. And I think continuing these public-private partnerships, developing new technologies, I think we need to continue to do those things. So when I say improvement, there's been a lot done there. I don't think we should stop. Regulatory and policy reforms, also well outside my pay grade, but we have done a better job with streamlining approval processes, but I think there's always room for improvement. And we do need to look at incentives for antibiotic development. They exist. What else can we do? If we lose, the brains in this room, there's a problem. And I think we all can acknowledge there's a problem, so what do we need to do to fix that? And then global collaboration strategies. Again, this is not new because there were multiple presentations before me that covered these as well. So for me, I went more with the pharmaceutical approach. What are the common pitfalls? What are we doing wrong? We're not always talking to enough people, right? So this interdisciplinary input is not always sought. We need to be having, top down, bottom up, left, right. Everybody needs to be involved in the conversation. If you're at a company, within that company, you need to be asking the right advisors about the right questions. It isn't just your favorite advisor about a question, and you need to be willing to listen to what the answers for that are. So again, we get late stakeholder buy-in. There has been some interest group driven targets, and when I say that, it's who at the company may have the biggest, something that they care about. Currently, it's what are the biggest indications. We're seeing some of this change as far as what the indications are to what is a long-term indication. Can I find something where we can use this for a longer period of time because that has a higher probability of financially being profitable? So, making sure that we're having all of those internal discussions with the teams that are working on these programs. Failure to revisit the data. You get new data, you should look at it in line with the data you had previously. No-go, no-go decisions should be go, no-go decisions. I have been on a program where we had a very specific go, no-go decision point, and we got there, and it failed, so it was going to be no-go, but somebody who had the bigger voice, the bigger in that room said, you know what, we're going to move it forward anyway. I could tell you it did fail. We didn't push it to phase 3, but it was, we'll do another phase 2 study, and it did fail. Now, you could also say, was our go, no-go decision right? What should we do? But in that instance, we had parameters. We didn't follow them. Instead of going back and starting with one of the others that was a follow-on program. Okay. So how do we do this better? And this has been brought up by multiple speakers today, and it's the TPP, right? So we need a target product profile. We need to understand what we're developing, why we're developing it, where we're developing it in, what is the unmet need in the market. They're not the same. Unmet need and market are not the same. So I recommend, and it's not just me, I think many people recommend, you have to have the end in mind, right? You can have an amazing compound, but if it has no activity against anything that's relevant, that means it's probably not something we should spend money on trying to push forward. So this is just, again, so what is the unmet need and how will you fill it? Is there a market, right? So what do you think your label claims should look like? And what label claims do you need for you to address this market? And can you get to those label claims based on the current regulatory status, et cetera? So we need efficacy, we need safety, but is that going to be logical for your market approval? Phase 3, what population are we really interested in? Are we going to include them in our study? Are we not going, and if we don't include them in the study, what additional data should we be generating so that we have data on that patient population that we're looking at, right? If you're looking at specific AMR, specific drug-resistant strains, you can't run a, non-inferiority, head-to-head study for those because the other drugs won't work. So what are you going to do? What additional data do you need? How are you going to get there? In phase, and again, we're backtracking into it. So, what does our first in human look like? And will that relate back to what we eventually want? And then finally, what are animal models that we're using to eventually get to? And what additional data are we collecting early on so that we can de-risk the program and understand that that market, that group of patients that we think we want to treat, that we're going to have the right data to fill that. So it is a key strategic planning document. And I guess maybe I'll ask, who here has actually worked on a TPP? Okay, good. So it's a strategic planning document. It provides a clear understanding of requirements, expectations of what you want the product to be. And this is multifunctional. You need everybody on this. You need your CMC. You need to know, do you need an oral? Is it okay if it's just IV? Is it reasonable to think you can make this product oral or IV, depending on where it starts, but usually it's IV to oral problems. So this is really, and then I put it on here, which may be a little, not a discussion point for today, but it's driven by the patient, the unmet need patient, the physician. They should be taken into consideration. What are physicians really going to be tolerant of? And what is really going to resonate with them? And then finally, the payer. we run into payer problems quite a bit, and we should consider what it looks like for them. Why would they pay more for this? And for who would they pay more for this? And is that enough for us to develop a drug? Now, so those are questions we have. So we need to have a roadmap with key go, no-go decision points. And you need to have a statement of overall intent of the product development program. Again, this is much more than just a draft label. You're not just going to put your dream label together. You need to think about all of these components. Again, like I said, CMC needs to be part of this discussion, but you also need your C-suite to buy in, that if this is what you're doing, this is what it's going to look like, and we've all agreed that this is how we're moving forward. So the good news on this too is, because I said it started out this way, there is a guidance document on your TPP, so you can look that up, and this is the specific one from the FDA. So it's there. You can look what they recommend, what their format looks like, et cetera, and what some of the examples are within there. Okay. So you need a differentiated value proposition. You need it's a snapshot in time, which means it does have to be revisited as new data comes in, not just internally, but external data, right? What are your competitors doing? If your competitor that you were most worried about that suddenly has a failed study, that changes where you're at. Or if they have a successful study and they were able to fast track their program, that will change likely where you're looking. This is a summary of studies that justify your labeling claims. So every single thing in that label is annotated to how you can say that and what data supports saying that. It's a communication tool internally, potentially parts of it to investors, potentially to others, right? And it is something that you can pressure test at advisory boards, et cetera. Do you agree? Do you think we can say that based on this? It's dynamic and multidisciplinary, and it is really a starting point for your annotated package insert, so your label. Okay. I stole this one off the Carbex website. I hope it's kind of fuzzy, so sorry about that. But really what it looks like, sorry, microphone. The minimal requirements versus your ideal requirements, or what your goal is. So what do you minimally need, and where do you think you could potentially get a win if you get something slightly different or greater? And then you'll have on the far left side the different things you're looking at. So the indication, which organisms, your patient population, your treatment duration, which does matter, right? Your delivery method, dosage form, regimen, efficacy, side effects, stability, cost, and other specific population claims are on there. There's also one from the World Health Organization, this one's specific for enteric fever. We worked on a draft earlier today of kind of what a TPP looked like. And it is a very interactive process. I don't know how many of you were able to make that earlier session, but people didn't agree. So we didn't land on one specific way, right? And particularly in that discussion was for pediatric formulation, what's acceptable? And so you really do have to listen to all of the stakeholders and have all of those discussions. So my advice is you establish your targets early, and I'm not saying your actual target for your antimicrobial, but with the end in mind. What is your target? If you don't know where you're going, you'll be lost when you get there. So where are you headed? It's an interdisciplinary activity. I really can't stress that enough. I think we often work in a vacuum, and I do think, I think it was mentioned earlier by Jennifer as well, sometimes specifically with anti-infectives, we're a small team, and we all wear all the hats, and we take new hats, so we need to make sure that we're looking a little bit outside of just us. You want to pressure test. There are different ways to do things that we may not know about yet, but we should learn. And then senior management should have input, and they definitely need to have buy-in. And then there should be a decision log. Somebody should be monitored. We don't have to revisit every decision all the time, or why it's there or why it's not there. So why did we make that decision, and has any data changed to drive us to make a new decision? Okay. So that's it on TPP. So Also not spending a lot of time on this. I didn't realize there was going to be a great presentation before me on this as well. But how do current guidelines fit the new non-traditional therapies? Again, my role in this particular presentation was IND to NDA. So where do these non-traditionals fit here? If I'm told you at the beginning, just follow the guidance documents. There are not guidance documents for a lot of these new non-traditional therapies. As mentioned before, you have antibiofilm agents. Well, how does that work? If we're doing it adjunct to existing therapy, you can do a superiority study, but is that really feasible or not? Is there enough incremental room to show improvement within those settings? If you're looking at virulence factors and being able to, decrease the effect of virulence factors by any of these new modalities, what is your endpoint? Because you're not decreasing the pathogen load, you're decreasing the effect on the patient. And so these are very different than what our current guidelines, guidance documents look at. So we are trying to put this square peg in this round hole and to try to understand. And I do think this has to be a conversation that continues within this group. But when I say within this group, this meeting was originally set up, and I think it's done a very good job. I mean, I was on the first program planning committee. We wanted to be a third regulatory, a third pharmaceutical, and a third academic. We wanted this to be a real collaborative meeting, and I think you need all components of that to move any of this forward when it comes to these non-traditionals. How are we going to get these to fit into a package that is approvable? Okay. Again, I didn't see other people's presentations ahead of time, but we do rely heavily on MIC, and zone diameters and susceptibility information may not be relevant for these non-traditionals. And if it's not relevant, or if it's kind of pseudo, you can do it, but it doesn't actually matter to the mechanism of action, should we be wasting time and money proving that we can't do it or that it's not relevant? If we can just say that based on the mechanism of action. Those are important things we should think about to decrease work that doesn't need to be done because it's not logical. And then if you're now measuring only clinical outcomes and not the microbiological outcomes, what does that look like? What does that look like for labeling? What does that look like for physicians and explaining to them the use? How, we'll talk back to the payers and the regulators, right? We have to come up with a conversation about that. So at the end of the day, to be successful, you need differentiation. We need it to be different, or else it is very hard to know where or how to use it. You need indications that make sense for what you're developing. We probably need new endpoints for that, and we need to focus on what we're doing. And I didn't add all the money market issues because I know that's going to be lots of discussions later. But I will end with one very, I'd say, disappointing slide. Is in 2024, there have been approvals, more than just these, but there were 3 antibiotics that were approved so far in 2024 that are not, in the US, that are not being commercialized. And those 3 companies have have mentioned that they are looking for partners to commercialize it, and they're not willing to do that on their own. They're not willing, they cannot put the money in to actually commercialize 3 approved drugs. So they've done all the work. They've been able to get through all of the steps, and now it sits. And we all know patent life is a problem anyway, but it'll just sit. So yes, we can develop antibiotics. I actually think that is not a problem. We know how to develop antibiotics. We're learning how to develop non-traditionals, but we do know how to develop antibiotics and antifungals. I don't think that's an issue. The issue is the cost and all of those other things, but we know how to do it. But if there's no market, or if there's no patient population that's easily addressable because physicians aren't changing their way of treatment, because yes, we agree that resistance is an issue, and many people are very worried that resistance will increase for these new agents, but meanwhile they sit and patients are not started on necessarily the right treatment first because we don't have the fastest ways of doing susceptibility testing. So there's a lot of issues when it comes to what are we doing, when do we do it, and that's on the back end, right? That is finally when we get to market, who's going to use it and what's going to change how they use it. Antibiotics coming to market do not get used regularly, and that's the data. So anyway, that's all I have.
Thank you, Judith. Subject very close to my heart as well. Thanks. We don't have time for questions at this point, but certainly there will be questions probably directed towards Judith as well as the other panel speakers when they join the round table at the end. So at this point, I'd like to switch gears a little bit now to get into the regulatory perspectives on antimicrobial development. We're going to start this two-pronged section with a talk from Dr. Radu Botgros. Dr. Botgros is an infectious disease specialist who has the position of Senior Scientific Officer for the Department of Public Health Threats at the European Medicines Agency, the EMA. He worked as an ID clinician for 10 years before joining the EMA in 2009 as a scientific administrator in the pediatric team. He then moved on to the anti-infectives and vaccines team, where he worked with the efficacy and safety related pre as well as post-authorization aspects of central marketing authorization applications for the treatment and prevention of infectious diseases. So since March 2020, he's been a member of the Public Health Threats Department. His main interests are not only with antibacterial agents, but with antifungal and antiviral medicines as well. So Dr. Botgros, thanks very much.
Thank you very much. And I'm very happy to be here to present to you what we at the EMA are doing with regards to drugs that are relevant for antimicrobial resistance. I'm very happy that we heard Judith's presentation before, because I was kind of worried if the audience had the same understanding of the process, but now I'm relieved that this is the case. So I think I will be able to transmit to you in a meaningful way something that often is perceived as being cumbersome. So on this slide, you will see the different activities that we are conducting at the EMA to contribute to combating AMR. And you see that we have a range of activities. I will focus today on the guidance to developers, which I thought was meaningful for a bootcamp talk. But you will see also that we have international regulators on board when we make our decisions. We also focus on alternative therapies, and I was very happy to hear today a number of times that there is interest for that. We believe we play a role in prudent use of so-called old approved antibacterials, and I'll allude to that in my talk as well. Then I would also like to convince you that we have regulatory tools at hand to allow that these very much needed drugs can make it to the market from the regulator's perspective. You heard today that this is not only the remit of the regulator, it's far more than that, but at least we do our part in a meaningful way. And last but not least, we are happy to say that we have in the past 5-6 years tried to engage with developers as early as possible in the process, knowing that most, as has been mentioned today a number of times, are small and medium enterprises. So, folks that may find this regulatory environment particularly confusing. So we have, and you will see later on in my presentation, for novel antibiotics, a forum in which we invite developers of such drugs to engage early with us and benefit from some sort of informal, if you want, advice before the general process of scientific advice that we have. Last but not least, as we all know, AMR is is not only about humans. It's also environment, it's also vets. We at the EMA, we don't do much on the environment because there is an environment agency of the European Union that has this in its mandate, but we have a vet department that approves including antibiotics for use in veterinary medicine. I won't be talking about that today, but I think it's worth knowing that we are engaging with them quite a bit as we believe that this should be seen in the One Health perspective. Here you see the main guidance on evaluation of medicinal products indicated for the treatment of bacterial infections. So this is our core guideline that has been revised recently. We used to have the guideline, an addendum to the guideline, things that had to be read successively, but also you needed to work with them such that it made sense. So we felt like revising this and compiling it together in one single document could be helpful. And we also revised a little bit content-wise. We have provided, for each and every major indication, what we believe are relevant development expectations of the regulator. And also we have a pediatric addendum that talks about developing these antibiotics in children. In that one, we also tackle the important subject of extrapolation that has been mentioned today as well. And we delineate those indications for which we believe extrapolation is possible for efficacy as well as for safety in certain conditions. We also delineate those indications in which extrapolation is not possible, for instance, in those diseases that are present only in children, to give one example. But also, we need to have clarity on what cannot be extrapolated from the beginning. For anything else, because obviously any such guidance cannot be comprehensive. We cannot cover everything that developers might wish to develop. We invite you to come to the EMA and discuss with us what your project is, so that we can have an early understanding and agreement as to what can and should be done to make it approvable. And we also have, quite important, you heard it today a number of times, a PKPD guidance. That's something I would also warmly recommend you to consult whenever you develop an antibiotic, because it's quite important. As I said, my main talk today will be about what is the pathway to develop an antibiotic that is relevant for AMR. And this is what we call the limited development program. You know that for a standard antibiotic, an antibiotic that is supposed to be approved in a standard indication, say urinary tract infections or intra-abdominal infections, the standard is to conduct 2 randomized clinical trials in a relevant population, with the whole adequately powered, all these things, and obviously the results need to support approval. So that's where we start from. The limited development program, what it does, it actually acknowledges what Judith said in the previous presentations, namely that it's not feasible to conduct trials that would enroll only patients with with infections produced by resistant pathogens, and therefore in this kind of situations, we, as regulators, we have decided to allow a limited development program. In order to even start thinking about it, you need to be eligible for that. So what does it mean? First of all, we need to, this eligibility, first of all, it should start from the developer. You will look at the criteria and, first of all, you need to find out whether you believe you're eligible. And then you need to come to the regulator and agree with the regulator that your thinking is also the regulator's thinking. Now, on this slide, you see that provisionally you based your thinking on in vitro microbiology and non-clinical efficacy. And obviously, you have to see that all that has been shown also on the previous slides, that human PK plasma exposures thought likely to be needed are achievable and that they can be tolerated. This is what you do in the early development. You see that PK and PD and the probability of target attainment are initially done using healthy subjects and later are repeated using infected patients. So in other words, you update the model with data from patients. And here I tried to dichotomize, you can have a new medicine of a new class, or you can have a new medicine of a known class. And you see here that for a new medicine of a new class, you need to demonstrate that the mode of action is unlikely to be affected by existing mechanisms of resistance, and you need also to assess the risk of cross-resistance, and that would include, obviously, non-drug specific mechanisms. Whereas if you have a candidate which is from a known class, you need to demonstrate activity against organisms that are resistant to other agents from the same class and understand the mechanism and frequency of resistance to the new medicine. You would also need, for the discussion with us, to describe the target pathogens, to establish that there is, in your view, still an unmet medical need. Here I need to stress that, you could have the situation in which, and now most of the candidates that are in phase 3 are beta-lactam, beta-lactamase inhibitor combinations, and as we heard today, they differ one from each other based on the combination, but important is to say that we have not stated in our guidance a number that, we acknowledge more than one such combination targeted that infection is useful to have, but at the same time, if you would have 7, 8, 9, 10 similar ones, maybe at some point you will have to consider that the unmet medical need is not there anymore. So I think what's important here, if you are aware that you have 3 or 4, and I'm talking numbers without any support from the guidance, it would be good to check that also the regulator still believes that there is an unmet need, because otherwise you may end up in a bit of trouble at the time of approval. From even today, I can tell you that as opposed to there are some differences between EU and the US, one of which is also about this unmet need. I think MRSA is considered, I'm speaking now under the control of my colleagues from the FDA, but we don't believe that MRSA and penicillin-resistant strep pneumonia are pathogens that are not covered by approved antibiotics. Therefore, these are not eligible or, for a limited development program. So I think that's important to say. And this reminds me of the fact that we have also another difference in the labeling. In the labeling in the EU, based on the program that was presented by Judith, you will get an indication that is generally not qualified by the pathogens. In other words, you would get something like treatment of complicated urinary tract infections in adults, let's say, and then we have in the summary of product characteristics, section 5.1 delineates the pathogens that have been we have also essentially, what has been studied in clinical trials, what is active in vitro, and so on. So that's also a difference, and I think for those of you that may not have done both processes, it's important to understand that, because many people wonder how is it possible that with the same development program, with the same dossier, the labeling is not exactly the same. It's not exactly the same But I think, this should not and is not, to my mind, any problem with developing the drug as such. And this needs to be factored in your TPP, as I understand that the labeling is part of it, isn't it? Now, coming back to the limited development program, you see that actually it's this the clean farm, and a strong PKPD support for the adequacy of the dose regimen for target patients and pathogens. It's something that we're required to see, and also speaking about safety, which has also been alluded to a number of times today. Here we don't have specific numbers. We generally think that the size of the safety database should be proportionate to the unmet need, to the expected benefit and observed safety profile of the drug. So in other words, again, come to us, to scientific advice, to make sure that whatever you will be proposing is acceptable. The clinical efficacy data will depend on the aim of the initial program. And here it's important to understand that your claim, your proposed labeling plays a role into, how the data package should look like, but also potentially in what will finally be approved. You see here that, and this I think are important things to mention, the evidence for activity against target-resistant pathogens will primarily come from micro and PKPD investigations. And you see that the RCT that you will be conducting will only, you could enrich the population for target MDR pathogens using rapid diagnostic tests if there is a suitable comparator. Otherwise, you can enroll, and you should enroll probably, a typical population for the type of infection under study. So that's, I think, an important message, that we are not requiring that you conduct a study in infections produced by MDR pathogens from patients. This I think is very important, and it sorts out the problem of feasibility. You can, as a developer, conduct a supplementary trial confined to or enriched for patients infected with target MDR pathogens, but this is not required if we believe the PKPD package is robust, and you heard today what robust means. I think that's also quite important to say. We can, as opposed to the general rule, we can and we actually do qualify the indications that can come out of the assessment, the pathogen-specific indication in patients with limited treatment options, if we know for sure that, for instance, there are penetration issues in certain tissues, or we could add a warning if there is any concern of such sort. So the good news, I think, is that in these MDR-relevant pathogens, we recommend one randomized controlled trial in one type of infections appropriate to spectrum. So you see that that's the limited development. Obviously, this trial does not have to demonstrate non-inferiority based on the margins that are, if you consult our guidance, you will see that for each major indication, there is a proposed acceptable non-inferiority margin. In this limited pathway, we don't require the non-inferiority margin to be achieved unless you have the willingness to also claim for a standalone indication. So in other words, if you have a drug that, say, works for drug resistance bugs that produce a complicated urinary tract infection, you could get the limited patients with limited treatment option indication, and I will give you on the next slide, I think there is an example of what wording of the labeling you could get. But if you want to claim the whole indication, treatment of complicated urinary tract infection, then obviously your study should meet the non-inferiority margin that is recommended. I'll talk a little bit also about the role of these clinical data in patients with infections caused by MDR pathogens. We heard, and we as a regulator agree on the fact that very small trials can be performed in this study population, and it can take quite a long time for the study sites to recruit. Therefore, also the studies that you can get can be difficult also to interpret and could delay approval and access to the patients in need. This is something that our guidance recognizes. On the other hand, as we also heard today, not only in the EU, the payers still want to see clinical data, underpinning the value of the drug. And so many developers choose to conduct this additional RCT in an enriched population just to have more evidence to show payers that this drug is of significant value. So all in all, I think it's desirable, despite the limitations that I mentioned, to gather outcome data in the patient population that is targeted. This is how the indication that we could grant could look like. So it's treatment of infections due to the pathogens that are targeted by the drug in patients with limited treatment options. And as I said, you could have a warning, you could have it qualified, like it's written below, treatment of HAP-VAP due to some types of pathogens in patients with limited treatment options. As I said, if you want to claim a standard type-specific indication like complicated urinary, obviously you have to have this one trial, at least one trial, to meet all the requirements that are provided by the guideline, including the non-inferiority margin, importantly. And then you could have the standard indication plus a pathogen-specific indication, as you see on the slide. Importantly, as was mentioned here, and I'm not going to spend too much time, PKPD, as I said, is very important in development of antibacterials, and here you see what are our expectations on the PTA that is supposed to be reported for the PDT associated with difference. You can have 1 log reduction, you can have 2 log reduction, you can have stasis. And here the guidance states that for potentially life-threatening infections and low spontaneous resolution rates, the PTA expected to be reported is for the PDT associated with 1 log reduction in CFU. Net stasis, on the contrary, is sufficient for infections which have lower organism burdens and for which important adjunctive treatment is used. And obviously, other aims can be considered when you select the PDT to be used in analysis of the PTA. And one of them, speaking about AMR here today, is a PDT associated with a minimization of the risk of selecting resistance. Here you see that it's generally expected that the proposed dose regimen provides a PTA of higher than 90% at the MIC 90 or epidemiological cutoff value, and that a PTA higher than 95% could be expected for regimens to treat life-threatening infections for which treatment is already available. So for all these details, I think it's good to get back to the guidance to make sure that you're conducting the right tests and studies. There are a number of things regarding PKPD aspects that we acknowledge can be further discussed, including with our fellow regulators from other jurisdictions. And you see here that, for instance, handling of heterogeneity in the PDTs which are derived from different models, in vitro and in vivo. Different labs leading to different results across the tested strains. Then, approaches for selecting the PDT to be carried on for PTA assessments, type of infection targeted, suitability of the non-clinical models, and so on. So there are still things that we believe can be further fine-tuned, and we are aiming to doing this. Here I try to give you an example of a new BLI, of a new beta-lactamase inhibitor. You know that this type of product can be partnered with a new or with a licensed beta-lactam, and it can be supplied in a fixed dose combination product, or it can be standalone with restriction to co-administration with specific beta-lactams. And I think this is the current situation, and the sponsor will need to provide evidence based on what the claim is. So here you see also that such a drug can be developed for either the site-specific indication or for a pathogen-specific indication, and that the BLI can have also intrinsic activity on its own, apart from the BL that it expands the spectrum of, or it restores, actually, better said. When it comes to microbiology, you see here that you need to look into the enzyme range kinetics, the MIC of BLI alone, and potentiated MICs of partner BL against strains. And so, also importantly, you need to derive the PKPD index and the PDT from non-clinical infection models using an appropriate selections of strains that are either resistant to the beta-lactam or are susceptible to the combination. Again, here we need to further discuss with the regulators from other jurisdictions a number of aspects, like, for instance, the role of the PKPD in streamlining clinical development, with the understanding that it cannot completely replace the need for clinical trials, but still, as you have seen, for a robust PKPD package, you could underpin the dose regimen, for instance. There are other things that you cannot, like, for instance, the treatment duration, to give one example. We need also to rediscuss to what extent these clinical data from target MDR pathogens are really needed. There are also other things pertaining to getting post-approval data in this kind of infections, like the role for global registries of infection that can be discussed, and we are aiming to tackling these points in the near future. And this brings me to the general interactions that we have with the international regulators. You see here that, first of all, we have, with the FDA since quite some time, a parallel scientific advice. So this is a review in parallel of the scientific questions that a developer has. This is discussed via teleconference, or it can be a written scientific advice. There can be oral feedback provided to the applicant at a meeting with the applicant and the two agencies. And also individual formal feedback can be provided to the applicant as an outcome. We have also another interaction, which is called consultative advice. This allows applicants to request scientific advice from one agency, one of the both, and concurrently notify the other one of the request. I think this is something which is also quite important. We discuss all the developments that are AMR relevant 4 times a year with these 4 agencies that you see on screen. So with the FDA, with Health Canada, and the Japanese, the PMDA. This is standard 4 times a year, but it can be done ad hoc whenever needed. We also had in the past trilateral meetings with the PMDA and the US FDA, where we have reached quite some consensus. You know, the aim here is to, as much as possible, come to requirements that can be applied independent of the jurisdiction, and we have been able to simplify and to agree, for instance, for urinary tract infection on the microbiological endpoint, which in the past was different, but now is the same. And we are aiming at resuming these discussions now in a quadrilateral format, because also Health Canada has expressed the willingness to participate. And this will happen, most likely the first one will happen early next year. Speaking about alternative therapies, you see here it's just a selection of what alternative therapies means. With phages, and I think this is something I would like to mention, phages, you know, I think I personally have read in a number of papers, and I have heard at various conferences that some colleagues that either develop or have interest in developing phage products believe that the regulatory framework is not appropriate. Now, this is only partly true. What is true for sure is that there is no guidance which is specific for these type of alternatives. That's correct. However, the principle of the general guidance still apply. That's number one. Number two, in order to be able to draft a guideline, to have a guideline in place, we need to see applications. And this is the funny thing, that despite, I think phages are one of the most, you know, I think they could be first on the list in terms of alternatives that could eventually get approved. We don't see all too many applications, and at least since 2 years, I have been inviting developers to come to us whenever they have such a product, to allow us, you know, to have an honest discussion and try to identify what the problems are and what we could do to facilitate. But as I said, until to this very moment, I personally have not seen, it's just a handful, unfortunately. Monoclonals, a bit better. We have approved some in the European Union since a few years now. We are also looking into other options, including what has been mentioned today, you know, starting from vaccines for, you know, here on the slide you see therapeutic vaccines, but also prophylactic vaccines as well. So we are very open to discussing with you, knowing that there is this absence of specific guidance, but at the same time inviting you and warmly supporting that you come to us to discuss, because otherwise it's very difficult to produce a guideline with little to no exposure to the development. That's one thing. And then the other thing is also that framework-wise, at least speaking about phages, I believe that indeed in case of personalized therapy, because, you know, phages can be administered as a predefined cocktail, can be developed as a predefined cocktail. For that one, the framework exists and is there. Whether these products will work, will show efficacy, and whether they will come on the market, is a different story. But the framework is there. For personalized therapy, I think we need some tweaks. We are discussing, you know, with the European Commission. You know that there is now a revision of the pharmaceutical regulations. So, you know, we have this in mind when having the discussions with our colleagues. So we hope to get also there to something that is feasible. But again, we need your input as well. We have this role in the prudent use of antibiotics. We have a tool. I won't be expanding too much on that, but essentially, the situation is that in the EU, all antibiotics have been approved nationally. So before the EMA, which is the central regulator, was ever existing, you know, so it's before 1995. And therefore, you have a plethora of SMPCs with different indications approved, with different SMPCs It looks quite dire, I have to say, whenever you have to start a procedure that we call referral. So in other words, the process of harmonizing and modernizing the PI is being referred to us, and then we start, reviewing all of these. And I can tell you, it's quite a lot of work, and you need to review all data because essentially it's a re-evaluation of the benefit risk. I think it's very useful to the practitioners, though, because I can tell you that, and you can have a look because the results are public on our website, but some of them really had different dosage regimens approved for similar indications, which, as you can understand, is probably not ideal when we talk AMR, but not only. So I think we're working still on this, and I think this is the most valuable contribution that we can have to prudent use of antibiotics. Obviously, we are not doing stewardship as regulators because we are not in hospital. Therefore, that is not within our mandate even. Here, and without spending time on each and every one, I have tried to list all the regulatory tools that we have for medicines in general that address an unmet medical need. The most, I think the most known is the accelerated assessment. And obviously, if indeed the drug is being shown as addressing an unmet medical need, and if the CHMP, which is our committee for approval, to make it simple for you, if they agree to this, then the accelerated assessment kicks in. In other words, instead of having a 210 days approval, you can have it approved in 150 days. This is based on the unmet need. If ever, however, during the assessments, the questions that are asked by the committee are not sorted out, then it could revert to a standard timetable. So I think it's important that the cooperation between the developer, between the applicant and the EMA is at its best to allow for this accelerated assessment timetable to stay. I will just jump into this early engagement with developers. As I was saying, it's something that we have started a few years ago. It's meant to be an informal dialogue on scientific, legal, regulatory aspects in a style of brainstorming meeting, if you want. But it's supposed to happen just for innovation. So, drugs, antibiotics that are innovative of some sort. So not for me too's. So to make it quite clear. But if this condition is there, the good news is that it's free of charge. You will get to talk to our experts, including from the network, and have this informal feedback, which I think could be quite useful for you to shape the program and then come for proper scientific advice at a later stage. Now, obviously, there is a template, there is a procedure for that, so that we have a bit of an evidence and we can try to guide those applicants that maybe, as I said, have some difficulties in understanding the regulatory framework, because you need to have questions that can be answered, so crystallizing the question is quite important. You cannot, or you should not start such a process, such a discussions with questions that we cannot answer for a number of reasons, be it lack of data, be it lack of understanding of the whole concept and so on. So what we are trying to do is to ask you to fill in a template with what your thoughts are, and then someone from the EMA will contact you in case there are any unclarities before this process is launched. And once this is clarified, obviously the meeting will take place, and we hope that this could be beneficial, and I think it has proven beneficial in many, many situations. I've come to the end of my presentation, so I think importantly we have these abbreviated clinical programs for drugs that address unmet medical needs. That's good to know. We have our new updated guidance to industry, which is since 2022 in place and is available on our website, so please consult it, including the addendum on pediatrics and the one on PKPD. Obviously, we do interact, rest assured, for all developments that are relevant with our partners from the United States, from Canada, and from Japan, to try as much as possible to agree on what can be agreed to make the development program as smooth as possible so that the drugs can come to the market as early as possible. So I thank you very much for your attention, and if there are questions, I'm happy to answer them. If not, maybe I don't know what the preference is. I don't have an overview of the time. I hope I didn't overdo my time. Yeah.
Next to the stage is Dr. Peter Kim. Dr. Kim is a director of the Division of Anti-Infectives in the Office of Infectious Diseases at the Center for Drug Evaluation and Research, or CDER, in the Food and Drug Administration in the U.S. And we welcome his presentation today. Dr. Kim.
Thank you. I'd like to thank the organizers for the invitation to come and speak today. Really appreciate it. And I know it's been a long day. If people want to take a deep breath and stretch, please feel free. Okay, so I'll be giving FDA's perspectives on AMR drug development. Let's see here. So ways that we help to hopefully facilitate drug development in the AMR space include, as our colleagues at EMA had noted, collaboration with other regulatory authorities, such as the tripartite meetings that led to alignment on the clinical trial designs for common indications and unmet need, as well as the quarterly meetings with EMA, Health Canada, and PMDA to discuss antibacterial and antifungal drug development challenges. And as Radu noted, we engage in parallel scientific advice at the request of drug sponsors. Additionally, we have an active pre-IND consultation program. Essentially, we ask people to come and talk to us early in their drug development, and we try to note or flag any particular issues that might result in a clinical hold, as well as give general advice on drug development programs. So we strongly recommend that drug sponsors utilize this program. In addition, we have some expedited programs, which we'll talk about in a little bit more detail today, such as fast track, breakthrough therapy, and priority review designations. Through the qualified infectious disease product designation, we provide some additional initiatives for drugs in the AMR space. And we do have additional approval pathways, such as the limited population pathway for antibacterial and antifungal drugs, as well as accelerated approval. Let's see. As everyone here knows, just to level set, we know that patients enrolled in clinical trials for new antimicrobial drugs often have life-threatening infections that need immediate treatment. It's typically the case that we don't have the micro lab results at the time of enrollment and treatment initiation. Yet investigators, sponsors, and physicians need to empirically treat. based on their clinical judgment. Therefore, in the clinical trials, we need to consider when patients are randomized to receive test drug versus active comparator, the trial would not be ethical unless one was to expect that both the test drug and the active comparator would be hopefully effective in the treatment of the patient's infection at the time of enrollment. Another challenge is, of course, the fact that resistant pathogens are rare, not only at particular clinical sites, but throughout the trial. And if we ever get to the point where patients have infections, many infections that are resistant to currently approved therapy, then we're in a public health disaster. Now, I'll provide a little bit of an overview on the expedited programs. They include fast track, breakthrough therapy, and priority review. As far as the qualifying criteria, a common theme is that the drug is expected to treat a serious condition, and then additional data would be required to demonstrate the potential to address an unmet need. For fast track designation, that may be non-clinical data, mechanistic rationale, pharmacologic data, and potentially clinical data. Whereas for breakthrough therapy designation, we do want to see preliminary clinical evidence that the drug would show a substantial improvement in a clinically significant endpoint. Regarding the timing of requests for fast track and breakthrough therapy designation, the sponsor should submit the request during the IND stage of development, and for the request for priority review designation, that should be submitted at the time of the original NDA or BLA submission or at the time of a new supplement submission. Timeline for response from FDA regarding these requests is within 60 days. Features include such items as rolling review, and in particular with breakthrough therapy designation, FDA would provide intensive guidance on efficient drug development. And for priority review, it allows for a shorter clock in the review cycle of the application. Of note, both fast track and breakthrough therapy designations can be rescinded if the drug product during the course of development no longer qualifies. Now a little bit on qualified infectious disease product designation. A QIDP is an antibacterial or antifungal drug intended to treat serious or life-threatening infections. Incentives include an additional 5 years of exclusivity for certain QIDPs, as well as priority review for the first application of the QIDP and eligibility for fast track designation.
Behind where you are.
I'm sorry.
Thank you.
Thank you. Okay. That's okay. That's interesting based on the display. Okay. So we were talking about the incentives by QIDP designation. The QIDP designation request should be at the time, any time before the submission of the marketing application. It should be noted that QIDP designated biologics are eligible for priority review, but not additional marketing exclusivity. Now, some points and highlights from the unmet need draft guidance from May of 2022. Candidate drugs are likely to have a new mechanism of action against bacteria that may have developed resistance to currently available antibacterial drugs. They could have an added inhibitor that neutralizes a mechanism of action, such as a beta-lactam combined with a new beta-lactamase inhibitor, or some other characteristic that has the potential to lead to enhanced effectiveness. Okay, so this slide is a wordy slide, but what it notes is that FDA has determined that it's appropriate to exercise flexibility in applying the regulatory statutory standards while preserving guarantees of safety and effectiveness for unmet need drug products, recognizing that physicians and patients may be willing to take on additional risk in the treatment of serious and life-threatening infections. Regarding the unmet need guidance, there are clinical trial considerations, both for non-inferiority trials as well as for superiority trials. From a non-inferiority trial perspective, it's noted that the active comparator should be effective therapy for the enrolled population. The clinical trial population should reflect the patients with the particular unmet need. We do recommend the randomized clinical trial design in order to have a comparative assessment of safety and efficacy. Okay. In addition, for non-inferiority trials, the trial could be based on a larger non-inferiority margin than is typically recommended in disease-specific guidances, but the acceptance of the NI margin would depend on the type and degree of unmet need. The sponsor would need to provide an adequate rationale, justification for that larger NI margin, and then we would discuss. Sponsors may also consider enriching for patients with the pathogen or pathogens of interest, and we'll discuss that a little bit more in the next few slides. And the primary analysis of non-inferiority should exclude patients with a baseline pathogens that are resistant to the control drug. So some considerations related to superiority trial design. Sponsors may consider enrolling subjects with more than one type of infection or body site with the pathogen of interest. Additionally, best available therapy may be used as the active control. The inferential testing statistical analysis plan should be agreed upon and discussed early with the agency. The unmet need guidance also discusses development programs for drugs that may treat a single species or a few species. Some considerations the sponsor would consider include the frequency of the bacterial species of interest causing serious infections, the use and availability of rapid diagnostic tests to promptly identify patients with the bacteria of interest causing the infection of interest, and the consideration of co-developing the rapid diagnostic test in the development program. Regarding the pre-market safety database, in general, a safety database for a drug targeting an unmet need population should be at least approximately 300 subjects at the expected dose and duration for marketing. However, the caveat is that the size may also depend on further characterizing any safety signals that may be noted during drug development. And the safety database can include subjects from all phases of development. As far as labeling considerations in a flexible development program, the labeling should include not only the known risks and benefits of the drug, but also a description of the limitations of available information that supported approval. And then in particular for drugs approved under the LPAD pathway, there are specific requirements regarding labeling and FDA review of marketing materials. As far as LPAD criteria, the drug would be intended to treat a serious or life-threatening infection in a limited population of patients with unmet need. Of note, the standards of approval still need to be met, and the sponsor needs to specifically request evaluation of the proposed drug under the LPAD pathway. We published the LPAD guidance in 2020. In it, we note that FDA may approve a drug under the LPAD pathway based on the conclusion of a positive benefit-risk balance in the limited population, even though there may be insufficient data to conclude a favorable benefit-risk in the broader population of patients. It should be noted that LPAD should not be used to salvage failed trials, and the required labeling statements help to ensure that healthcare providers understand the limited population of patients for whom the drug is intended, as well as the limitations of the data that led to the approval. The next set of slides include the drugs that have been approved under the LPAD pathway. The first, on particular for this slide, was Arikayce, approved for the treatment of MAC lung disease in patients with refractory disease. We also have Prudent approved in adult patients with XDR-TB or MDR-TB, in whom the patient may be intolerant or non-responsive to standard therapy. And DefenCath, which is indicated to reduce the incidence of bloodstream infections in adults with kidney failure receiving chronic hemodialysis through a central venous catheter. So some closing thoughts. If you remember nothing else from my presentation, I would just ask that you consider coming and talking with us early in your drug development, and to consider utilizing the pre-IND consultation program. It allows a large group of us from a variety of disciplines to evaluate your program, and we hope to provide useful feedback to you before you submit your IND. And then, of course, globally, regulators remain focused on drug development for AMR. Thank you for your attention.
I think we have time for a couple of questions to Dr. Kim directly, if there are some. And once those questions are asked, we could get the rest of the presenters on stage for the round table. So, Dr. Patterson.
Singapore. There's I guess a concept that entwining antimicrobial development with anti-cancer drug development may be a positive thing for development of antimicrobials. In the old days, there was an indication for febrile neutropenia. For example, I think cefepime has an indication for that. And in the last 15 years, I've not seen any antibiotic go for the indication of febrile neutropenia. Can you explain why that is? Is there still such an indication as treatment of febrile neutropenia?
So, Dr. Patterson, thank you for your question. Actually, we actually just had a workshop on drug development for febrile neutropenia just a few months ago. What we heard loud and clear from the panel was that a specific indication for empiric treatment of febrile neutropenia is not necessarily needed, that people would be happy with and have confidence in a drug that, for instance, was approved for HAP-VAP or complicated UTI. I mean, we're happy to reconsider that, but that was something that was brought up during the workshop.
Okay.
And I really appreciate the question because we've been thinking a lot about this particular indication.
Thank you.
Hi, thank you. I'm Ioannis, academic doctor from UCL in England. Both you and the EMA speaker mentioned that it's difficult to do trials in CREs, for example, because number of infections is low, yet we have labels for treatment of gram-negative infections with limited treatment options. And we talk about an AMR pandemic. We are in Southern Europe. There are hospitals here who have 500 bacteremias per year of CREs. So why are we not demanding trials in settings where those drugs will be used for the infections that will be used and that they get the right labels?
I think you have an excellent point, and I'd love to see trials in those areas.
Thank you.
Ultimately, sponsors decide where they're going to do trials. I mean, we can make recommendations as regulators, but ultimately, it's the sponsors that are paying the bill for those trials.
Dr. Kim, regarding the limited population pathway that you discussed and provided some examples for, do you think that the pathway as it's currently designed has been optimized, or would there be adjustments or revisions that might encourage sponsors to come and make additional submissions via that pathway?
I think there's always room for improvement. Again, part of it is Congress developing the laws. We're open to additional thoughts. There are certainly other sponsors who have been interested in LPAD, but drugs are in development. So, but certainly if you have additional suggestions, we're happy to hear them.
Thank you. So if we could ask that the speakers from earlier this afternoon all gather and into the chairs here, we'll start the roundtable session. Thank you. And while people are assembling, if there are additional questions that you've written down on cards or wish to have those read out, please look for ESCMID staff to collect those. Alternatively, you can just bring them up during the session.
Do we know if Tomas is type that?
Yeah, and perhaps one question for the AV specialists. Do we know if Dr. Thomas Doyle is online or can join us for the roundtable? Okay, until that time, perhaps we can begin.
Sure. Oh, okay, we have audience participation. Please, Ryan, go ahead.
I don't know the rules. Apologize. Ryan Source from Rebogenics. So I had kind of a two-part question following the last session that I'm interested in. Anyone's opinion, but Radu and Judith, I'm picturing you're probably most likely. So Radu, if a developer is currently only developing in the United States and has no current intention to seek approval in Europe, can you still use that consultative advice vehicle to at least keep EMA informed of FDA discussions, or would it be bizarre to use that mechanism if you're not really currently intending to seek approval there? That's your part, and then Judith, I got it.
Yeah, can you hear me? So I think it's important that as I mentioned, we have already these 4 times a year discussions, so there is a high likelihood that if it's something relevant, we may have heard of it from the FDA colleagues. And, this is probably something that you need to know. But obviously, if you would like to inform us, please feel free to do. I mean, it cannot harm for sure.
Yeah, and related to that, I'm curious, Judith, one of the reasons now in the kind of reset after all the bankruptcies is you have to be very cautious about what you kind of start down a path. And so in the past, adding Europe to an otherwise US-only approval added significant cost and complexity to the development program because of the key areas where there wasn't alignment. There's a couple in the CMC area I can think of that are real deal breakers, but I'll give you a pass on that because your comment earlier. So in the non-clinical, clinical, Judith, from your experiences, where were the major cost drivers of adding an additional region to an otherwise US-only approval?
So you're asking me? So obviously it's indication specific, right? Because there's certain indications that are a whole lot easier to add sites to than other indications. And it's, as you said, the cost issue does become adding countries, which is a huge deal to any kind of clinical trial scenario. And currently, I'd say one of the bigger costs with clinical trial work is the difference in IRBs, the difference in the data that is allowed to be collected. It's even, ridiculous on the microbiology side. It's hard to now align what you're getting and what the central lab is getting and how we're trying to align the numbers so that it's the same because of confidentiality differences between Europe and US. So it does add significant complexities when you do open ex-US sites. And I do have to say currently, clearly some of the war areas are causing issues in finding sites that historically have enrolled well as well. So those are huge issues that are different. As far as the actual dossier, when you're all said and done, not huge differences, you'll need a different label, which that's fine, that's not actually a huge difference. Those have aligned reasonably well, though the PKPD is more rigorous coming out of EMA versus FDA historically, but if you do the rigorous PKPD to start out with, which most people are doing due to trying to de-risk a program, that's not a huge deal. I'd say it's really getting to the sites, differences in the data that is allowed to be transferred, and every site is expensive, right? And Northern Europe is very difficult to figure out what to do versus Southern Europe, but then in Southern Europe, there are rules when it comes to enrolling patients where the patient has to give consent. And if you're doing a HAB-VAP study, that patient will not be able to give consent, and it's not a family member. So those are differences that will adds in possibilities, it does, and therefore it costs, right? If you're trying to open sites in all these places to have a minimum enrollment in certain sites. Oh, yeah. Oh, the pediatric, yeah, well, yes. Oh, well, absolutely, your PIP is very different, right? Your pediatric plan will become very different requirements.
Okay, Alice, and then I've got a whole list of cards here.
Hi, excuse me, Alice Irwin, independent scientific consultant. And my question is about alternatives to antibiotics, antivirulence agents, where we often hear, it's kind of a meme you see in research proposals, that resistance will not be an issue because they don't actually inhibit growth of the bug. But so thinking about Thomas Doyle's talk, it was clear, and thinking of FIMH, you're looking for resistance, and you know it's not going to be resistance in terms of an MIC, but I'm wondering, how are you assessing loss of susceptibility, which I'm sure happens for some antivirulence agents. It's hard to see why it wouldn't, like an anti-capsule drug is going to select for resistance. But anybody, you know, so specifically about the FIMH drug, but then in general, how do we think about resistance for potentially different types of antivirulence agents?
Tomas, do you want to?
Yep.
Yeah.
Can you hear me okay?
Yes.
Yeah, great. No, it's a great question. And I think that we should be careful about, you know, these broader claims that we don't expect to see resistance, because on the face of it, while we're not killing the bugs with the FIMH antagonist, of course, we are selecting on the population because you're essentially removing the bacteria from the population who are being bound out and urinated out with the FIMH antagonist, and yet there's others sort of hanging around. And if they were able to somehow evade the mechanism of action of the drug, then you'd be selecting for that population. So we are looking for this. Actually, we've tried to do a number of innovative things in the preclinical space, but we are looking for it also in our early human studies. So I can't disclose everything that we're doing, but obviously any functional assays that we have where we look at the potency of the compound, then we're trying to apply them in the clinical space. And I've indicated earlier on that I think that, you know, genotypic and sequencing-based techniques will be really important here. And, you know, we all know how to implement those against, you know, when we're looking at pathogens in a clinic, you can do it on a target-specific way, and you can look at whole genomes if you want to, and so on. So those are the types of, you know, approaches that we'll be using. But in general, yeah, I think, you know, one of the things I've been thinking about in preparing for this, but also listening to the talks, is to make sure that, you know, we don't lose the understanding of resistant susceptibility from the traditional antibiotic space as it applies to these, you know, antivirulants. So if we're going for single targets, is that an issue? Should we be using combinations? All this sort of thinking. They're new agents, but some of the same principles may apply. So yeah, thanks.
Much for organizers for organizing this very important, interesting conference, and to all speakers was very interesting presentations. As I am a clinical pharmacologist, I have one question about developing new combined antimicrobial drugs. Specificity for development, because we have a lot of experience in the past about combined antibacterial drugs, and I am interested about new combined antibacterial drugs, antibiotics. If you have some information, thank you. New combined antibacterial drugs.
And let me just make sure I understand. So by this, do you mean, for instance, beta-lactams with beta-lactamase inhibitors?
About development, about new drugs, new combined drugs.
So two NCEs combined together.
Yes.
Yes. I think that our guidance is actually designed for you know, indicating what our expectations are for new antibiotics. So I think if you have a combination, it very much depends also what type of combination it is. You know, any combination to antibiotics, why is it meaningful to have them combined for the indication that you're targeting? In other words, data that you have generated showing that there is an added value. In other words, you know, that synergism actually means that the value of the combination is higher than the value of the individual components separately taken. So we would expect, and the guideline is actually having a subchapter on that, at least in Europe. I can't speak for the US.
Yes, one more additional question. I asked about information about combined drugs which are undergoing clinical trial phases.
Well,
If
the
you have
guideline
some
does not only
information.
speak about clinical trial phase. It actually is, about the whole development, starting from the non-clinical part to progressing to phase 1, phase 2, and
Yes,
clinical.
yes,
So it's actually meant to, to address the whole spectrum of development, not only the clinical trial part.
Okay, thank you.
Okay, while you have the mic, I'm going to ask a question from the card, and I'm going to embellish it a little bit because I had a similar question. But when you talked about what is eligible to go down a limited pathway plan, you specifically called out a few pathogens that you didn't consider today an unmet need. And the question here is, why? And the spirit behind it is, what is here today, in 10 years, you may want something new, either because resistance is developing or because the performance characteristics of the products you have today may not be ideal. In your example of MRSA, many of those products are IV. Would a safer oral MRSA agent today be attractive for development under that pathway?
Thanks for the question. I think the reason for us having this thinking as of today is the fact that there is quite a significant epidemiological difference between what is, for instance, CA-MRSA in the United States and in Europe. We don't have the same situation, the same epidemiological situation. That's number one. And number two, we believe that at the moment, the need is covered by existing antibiotics that are approved. What you're saying, the fact that things may change in the future, of course, is a very valid point. I'm not saying that in 10 years from now we may think the same. It's just that at the moment, if you would come with a development for a limited for patients with limited treatment option for an infection cause produced by MRSA, most likely we will not agree with it.
Yeah, no, I think that the comment is, isn't it important to stay ahead?
Well, it is important to stay ahead, but at the same time, we need to have principles that are fairly applied to each and everyone, isn't it? As I was saying, like for instance, in those situations when you have BLIs that start to get approved in the same, so to say, setting, there may be at some point the thinking that maybe that's at some point the unmet need is covered. So again, these are things that we apply based on the current situation in terms of what is approved, what is covering what, but also the epidemiological situation, because obviously, as you can understand, if the situation is much more relaxed in that sense, I think that also plays a role in the whole judgment as to what is actually an unmet need to start with, and then about whether a drug covers or not that unmet need.
Okay, thank you. I'm going to ask one more question, and then I'll let you. You want to follow on to that? Okay.
Question to the regulators. So that's gram-positive. Oh, sorry, Paul McGovern, Venatorix. So a lot of us develop drugs on the gram-negative side. And so, we have the CRE out there. ESBL is there too. So does an ESBL-enriched trial get us somewhere in the limited population pathway in Europe or the LPAD in the US? I mean, we can go to CRE, but we're going to find less of them. There's a lot of prevalent ESBL out there, or does ESBL only apply to, as Aaron was inferring, just to the oral drugs because we have now enough IV antibiotics?
Right. I think what's important to be said here is that, I just delineated those two pathogens knowing that the question could be followed up for other pathogens. So I won't be able, to provide a definitive answer, this yes, this no. It's just a matter of, number one, what is your claim? What is the indication that you are you only seeking pathogen-specific indications? So that's quite important, whether you seek the whole indication or the pathogen-specific only. And then it's a matter of how you argue in your dossier as to what is the added value of the drug that you're developing. In other words, to be able to make sure that the unmet need is covered or is still existing, we need to know precisely what Judith was mentioning about the labeling. What is your drug developed for? Because maybe it addresses a niche that is not addressed by previous similar drugs, case in which it could still qualify.
It kind of creates two lists, though, however, because you have, or maybe a third list, because you have the CDC priority pathogen list, you have the WHO priority pathogen list, and now we have the regulators saying there's a list that we consider to be still unmet.
Right. I think it's a matter of, all these lists, you're right, they are not one and the same, that's correct, but at the same time, we are all aware of all of them, and as I said, for us as regulator, what's important is the benefit risk that needs to be positive in the indication that you're claiming, and for that, we need to be reassured when you make your justification as to why your drug is covering an unmet need. And I would imagine that it would not be just based on a list, isn't it? I think if I was to write such a justification, I would not say, oh, it's on that list or on the other one. I would say, look, this is the mechanism of action, this is how it addresses, these are the pathogens that are being targeted. And then, obviously, as I said, we are in a better position to understand and to quantify to the best of our knowledge the unmet need, I think. So I would not be, if I dare say so, very much impressed of the fact that there are different lists. I think these are valid in the context in which they have been generated, but I think the important bit is to make sense from the justification that you will be providing. I think this is also showing that we are not thinking in terms of lists only. So I think it's a discussion, and obviously it's the burden of evidence, of showing and supporting your claim is on you, but that doesn't mean that we think in terms of lists, in particular since the lists, as you correctly quoted, are not always the same. I hope
Dr.
at least
Kim,
I have
did
been
you
able to provide some reassurance that we are not just applying lists and, yeah.
Okay, so is this on? Okay. So I think you bring up a number of good points. One, I think we as regulators understand that the drug pipeline is fragile. Two, though in the US we do have a list written into the legislation, we're not limited to that list for consideration of, say, qualified infectious disease product designation. And then three, regarding LPAD, that's more about the amount of data that you have in your application, safety and efficacy, than about whether necessarily your drug would treat a specific pathogen. So certainly, if you were able to do a full trial for the particular body site and include pathogens, some of which may include carbapenem-resistant enterobacterales, we would likely recommend, if you have the data, to go for the full indication. But again, it depends on what we see in the application. And then, as was noted in in the prior talks, specific information on activity against those carbapenem-resistant enterobacterales would be in section 12 of the labeling.
Okay, I'm going to give the regulators a break. So I think this is going to be for Jen and Claudia, whoever wants to answer it. And the question is, do you think the lack of strength in the AMR field has more to do with the problem of biology? Specifically, it's not one pathogen but several, lack of good diagnostic, and many disease presentations.
Yeah, so actually, I gave a talk probably 6 years ago at ACS, and I used an analogy of basically when you build an antibiotic, you're building sort of like a Mars rover, but one that has to land on all 9 planets. So you are typically looking at a medically important spectrum, and a single pathogen, approach is appropriate in very, very few indications. And then, you're also looking at typically one or more indications as well, because if you have if it's a systemically delivered antibiotic, then likely you're going to want to ultimately treat more than one indication in order to have that label and that ability to market. So it does add complexity, because there isn't a sort of single consistent rule about how molecules attach to surfaces, enter bacteria, stay in bacteria, all the problems that you have with one pathogen, you now have to deal with those problems across a medically important spectrum. So yes, it does add complexity, and as areas like oncology get more and more precision approaches, in the antibacterial space, we don't really have that, like I said, that many examples of where that's a medically important approach. I mean, you want to be able to cover a spectrum. If you have a gunshot wound to the abdomen and you have an E. coli specific drug, what are you going to do with that?
I agree with what I heard. I'm not sure much I can add to the extensive, but yeah, let's say that of course some species, so the biology for some species, like the example of Acinetobacter, are species that we have drugs, but we don't have, is one of the species that is less studied in some way. So we cannot underestimate the fact that some species, now in the specific example, but we can have also other, where the biology can play a role. And then it's not that we are in the space of the non-traditional, but we have to be open to understand that we may have challenges that we don't have, and the biology, of course, play a big role to understand what is happening. So when we see some behavior that we cannot explain, for example, simply with the exposure of the drug in the site of the infection, then we may say what is the reason behind. And that's the reason why one of the questions that I put there was about the discussion. I don't know if we learn more during this conference, is about what we can understand also of the biology of the bacteria in the site of infection. What is really happening when we are there? How is behaving a certain species in the lung of the patient? And so I think if we can also improve in this direction would be beneficial for sure.
I think that's great. And before I move on, I want to invite Tomas back, because to your point about what goes on in the site, I think one of the things I saw on one of his slides was that they're looking at what's in the microbiome, in the local microbiome in their phase 1B, and I think that speaks to your point. So did you want to say anything about that?
What we're doing in the microbiome in general?
Mm-hmm.
Yeah, throughout our program, we're going to try to, first of all, understand, because, we're seeing this in humans for the first time. We already have a pretty good sense from our first time in human study of what happened. Now we're in patients, and we may be going into patients for more prolonged durations, where we'll be able to see the effect of the microbiome. But I think it's important as part of this antivirulence story to demonstrate what we mean that, when we say that the drug is antibiotic sparing. is microbiome sparing. So we may have some studies where we're able to compare the microbiome effects with that of antibiotics, even in the current study with nitro, which has fairly limited effects on the microbiome. But yeah, that's something that we're going to bring all through our program. I think we've already heard from regulators that they think it's important, and external experts, medical experts feel that, you know, to understand properly this class of agent, it's really something that they would like to see.
Thank you. Okay, back to the regulators. So, and actually, I think, you know, this is a great question, and I have a feeling I'm going to see David Patterson leap up again. But, you know, one of the things that we didn't, you know, focus on here, and I guess we can't focus on all things in as many hours as we've had, but the topic of gaining approval for drugs beyond the geographies that we've just spoken. And so this question is, would harmonization of regulatory requirements, especially with developing nations, for instance, India, encourage clinical trials in those countries and bring down the cost of development and therefore access to new antibiotics?
I think, thanks for the question. Well, I think the short answer is yes. To elaborate a bit, the regulators, we have a forum which is called the ICMRA, the International Coalition of Medical Regulatory Authorities. And this is probably the most important forum in which we can take this forward, and actually our mind is so to do. We have not only India, but also other important regulators there, you know, TGA from Australia, we have the Brazilian one, the Anvisa, and other regulators. Obviously this is a bit the next step that we are minded to, you know, to make on this avenue. So it's highly likely that we will use this forum, and actually this is part of our plans for the coming years. Harmonization of the development program means, you know, to identify the low-hanging fruits and to, you know, just agree on having them harmonized. We know upfront that there will be situations in which that, for a number of reasons, may not be entirely possible. So I don't think we should raise our hopes as high as to say that it's just a matter of agreeing, because there are differences in the legislation in the different jurisdictions. But apart from that, simply seen from the scientific perspective, I think we have already identified items where we can converge, and we believe that there's quite some merit in having those countries that you mentioned and others on the same effort.
Yeah, I would agree. I think this is on. Okay, there we go. Yeah, I would agree. I mean, certainly those are areas that may have high incidence of AMR type pathogens. We're more than willing to engage. This is actually something that we were actually talking about in the office recently. But we're happy to explore and interested.
And perhaps just to follow on to that, could you help us understand any advances with endpoints that are, for example, these standard core sets of clinical outcome assessments or DOOR type of endpoints, desirability of outcome rankings? Because it certainly, from what you said, Radu, it certainly seems like this might be the perfect opportunity, especially across countries and regulatory bodies. Surely people can all get behind the fact that they want their patients to get better, feel better, and survive. And so accomplishing those things, perhaps, you know, could this help resolve some of the very individual specific challenges about endpoints between agencies?
I think, thanks for this comment. I think the first step will be to include endpoints like DOOR in our guidances, because they are not there at the moment. So it's something that I think we are quite open to discussing, and, you know, reconvening the, you know, the panel that has been involved in in developing the guidance. But we need to be reassured, obviously, that this is, and I think there have been publications, recent publications, that have shown that it's working, so that's encouraging. But we need first to include it in our guidance, and then obviously we can expand to, to discussing it with other colleagues from other jurisdictions.
From FDA's perspective, specifically regarding DOR, we have been actively engaged with ARLG. We've had recent publications as well, actively exploring DOR for specific indications like complicated intra-abdominal infection, HAP-VAP. At this point in time, at least from FDA's perspective, we would consider DOR endpoints as exploratory in clinical trials. If sponsors are interested in including DOR endpoints in their trials, again, we would recommend that they include them as exploratory endpoints until we get additional information and usage of, and see how they actually perform compared to our current proposed primary endpoints. And then related to clinical outcome assessments, we are also actively engaged in research in several clinical outcome assessment tools. And again, how patients feel, function, survive, that's a clinically important endpoint that we would be very interested in exploring for different disease indications, if that helps.
I think that's great. Well, in terms of people feeling well, functioning well, and surviving well, I'd like to thank all of you very much for your presentations and engaging. And I think we can take the burden off and go to the reception and continue the conversation there. So again, very appreciative. It's a great way to start this meeting. Thank you very much.
Thank you.